Hello, hello, hello. Welcome, everybody. Welcome to the stream. Hello, it's been a bit of time. Welcome to you all. Hello, hello. Thank you for showing up to my post-Ebola video stream. Of course, as is tradition on this channel, I will be going into a little bit more mechanistic depth in terms of what we covered in that video. I'm very excited to share this paper with you, this paper on the specific mechanism by which Ebola kills T cells without infecting them, of course. But first, I'm going to go ahead and say hello to you all. Hello, hello, hello. Welcome, Silly Catboy and Archie and Otto. Hello, Gluon. Hello, the Skype Converser and Cyrix and Kitsune and Nothing. Hello, welcome. Mylid, welcome, welcome. Monish Rules, hello. Kalo 007, hello. A, welcome, welcome. Hello, Trojnet. Leo, welcome. Hello, hi. TK, hello. Eric, hello, hello, hello. Eirik, sorry. Hi from Norway. Good to have you here. So fun and lovely to have an international audience, which is a decent segue for me to talk about an announcement I have, which is I will be covering glioblastoma, an aggressive form of brain cancer, in a 12-hour charity stream that I will be holding on the 11th of January for St. Jude's. You can find the commands in the live stream title if you want to learn more, but basically the reason why I love St. Jude's Research Hospital so much is because they do have an international appeal. Not only do they provide cancer treatment for free at no cost to families for American families, but also they are investing in a global network of supply and distribution chains to get cancer treatments to patients in need all across the globe, especially right now in low and middle income countries. So I wanted to find a charity that would make everybody feel good donating to, and I landed upon St. Jude's. So please look forward to that. If you have the time that day, I would highly appreciate your company then. Hello, Mandrake Flower, welcome, and Jareer Money, welcome, welcome. All right. So, let's not dilly-dally too much. Let's maybe just get right the heck into it. Let me go ahead and move myself into the corner. And let's talk a little bit about Ebola, shall we? So, let's go ahead and start with just looking at the title of this paper. Ebola virus glycoprotein directly triggers T lymphocyte death despite the lack of infection, from my beloved PLOS journal. So it can be kind of tricky, I think, to pick papers for a stream. PLOS is a lovely journal where all of their content is open access. And I believe at least every single article that I've ever seen has the broadest level of copyright protection. Not the broadest level, but rather the most lenient level of copyright protection, allowing me to talk about this on stream, which I am, of course, very, very grateful for. So I chose this paper because, in actuality, this is something that I had learned after I published the Ebola video. So if you recall, in that Ebola video, I had said that T cells die due to inflammatory signals, which is true from what I can gather, but also Ebola mediates a specific type of killing of T cells that I haven't really seen in other pathogens. And so it's kind of exciting and fun to get to explore this field with you all here today. So let us get right into our story here. So I'm not going to give like an intro to Ebola, because hopefully you watched my Ebola video and that has all the reasons why you should care about Ebola. But one of the more particularly striking features of Ebola is this widespread death of immune cells. So I contrasted Ebola with the rabies virus in this video because I think that contrast is the most apt, right? Rabies is a virus that infects neurons. And rabies' whole thing is that it is very, very good at staying as quiet as possible for as long as possible, right? Rabies, if you remember that video, does things like prevent neuronal apoptosis, preventing neurons from sacrificing their own lives to stymie the spread of a viral infection. Instead, what rabies does is to keep them alive, even though their cell bodies are basically telling them to sacrifice themselves for the good of the body. That's not to say that rabies is completely quiet. Of course, once it gets to the brain, it can induce apoptosis in T cells, but the modus operandi of something like rabies is to stay undetected for as long as physically possible. And so Ebola, by contrast, does not appear to share that same strategy, right? So if you've watched my video, you know that Ebola is an explosive kind of virus. It gets in, and there's a bit of initial immune evasion and immune suppression, but once it gets rolling, it explodes. It is an incredibly violent disease, violent inside the body, causing all sorts of manner of tissue damage and death in all sorts of different cell types. And T cells are one of those cell types. Unfortunately, I was not able to show the data because the data that this comes from a paper where the copyright holder does not give broad permissions to republish, so I unfortunately can't show that. But the idea here is that Ebola is killing T cells in large numbers, but it does so without infecting them. Hey, Clockwork, welcome. So the question is, how does this happen? So the approach that these authors take, I like to call just casually a sort of bisection approach, right? So A asks all T cell types. All the T cell types that they tested in this paper, they've tested quite a few in terms of different lines of T cells. But so there are regulatory T cells as well as killer T cells, and then there are like subdivisions of those that I'm not super familiar with. But it does appear to be a broad T cell death with regards to Ebola. So they're taking something like a bisection approach where, you know, you're asking this question: how does Ebola kill T cells if it doesn't infect them? And so you can come up with all sorts of very specific hypotheses to test, but when you are just starting off investigating a certain problem, it can be helped to sort of bisect all possible situations. So no one else calls it the bisection strategy except for me, but it's just something that I'd like to point out here, is that there are two hypotheses that cover essentially all possibilities. Either Ebola must be causing T cell death directly, or it's got to be indirect, right? So you can either be one or the other, or maybe a mixture of both, but all possibilities lie within this definition. And so constructing experiments that test for this broad net allows you to rule out a lot of hypotheses right off the bat, which I think is a smart move. So under indirect mechanisms, this is something that I have actually covered in the Ebola video. So we have this idea of a sea of fire, this inflammatory signaling that's ramping up in the body, cytokine storms, causing apoptotic signaling and all the like. And then there are direct mechanisms. So it might be worth a little bit of time to sort of think about what a direct mechanism could be. So I want to give chat just a little bit of space to think about what a direct mechanism of Ebola killing of T cells would look like. Ebola is the internet version of regular Ebola, so my hypothesis is that Ebola makes mean comments towards T cells, which makes them melt down in a big Twitter thread. Oh, if only the T cell could meet such an end in such a humane way. Let's just put it that way. So chat, what do you think could be a way for Ebola to directly cause T cell death? So none of this sea of fire surrounding the T cell nonsense. What could potentially be happening? And, you know, if you're new to if you're new to my live streams or new to the channel, you don't have to have biology background. It's okay to just hypothesize at whatever level you can engage the biology with. That's sort of the fun of these streams, is that you can kind of test your intellectual mettle without actually having that much biology background. Production of antibodies that make them clump together. That's an interesting method of doing so. So make who clump together? Because we're talking about T cell death here. Cell lysis via viral replication. So that is not going to be the answer, because Ebola does not replicate in T cells. So Ebola doesn't infect T cells at all. Trigger apoptosis. Certainly, that is one possibility, triggering apoptosis. What about how is it going to trigger apoptosis is sort of more my question, because apoptosis is a very broad, widely applicable system that needs to be triggered, and we're talking about what triggers these mechanisms of cell death. Activation of toll-like receptors. Yes, so toll-like receptors are receptors of the innate immune system. These are little bits of proteins that can detect things that pathogens like making. And one of the sequelae, fancy term for consequence, of TLR, toll-like receptor activation, is cell death. That's certainly one possibility. But how are those toll-like receptors activated is another question. I remember seeing somewhere that the Ebola glycoprotein has a part called a mucin-like domain that just kills things randomly. I actually don't know about that. That didn't come up in my research. It'd be interesting to continue to look into that kind of thing. Immune exhaustion. I'm gonna have to be a little more specific than that. Just widely released pore-forming proteins. So that is more of a strategy for eukaryotes to fight pathogens. So that's something that we do to bacteria when we form the attack complex. Not something that I have heard viruses doing, although I'm not saying that's impossible. It's just not a technique that I've heard viruses do, which is interesting. Most T cells apoptose after an infection. It could use it that way. So Ebola doesn't infect T cells. Like Miansert, I'm actually unsure what that is. The best part of your channel, you explain stuff so well that people like me who don't have biology background can understand it. I'm glad to hear that. I think biology is extremely accessible. It's just stories of little guys after all, and everybody can appreciate a story of a little guy. So these are all interesting hypotheses, but I'm looking for something much more broad. And so in terms of mechanism, something like this would be a little more appropriate in terms of bisecting all possibilities, right? So maybe Ebola makes something that interacts with T cells and that causes them to die. So certainly a lot of answers from chat could fit under this definition, but in terms of a testable bisection hypothesis, this is something that is more easily testable. So it's making something that interacts with T cells directly, causing them to die, as opposed to infecting macrophages, causing them to die. Macrophages release a bunch of pro-inflammatory cytokines that causes inflammatory stress, which then kills T cells, right? You see how Ebola directly making a protein that touches a T cell, that's a direct mechanism, versus something indirect where it has to do a bunch of steps before the T cells die. So that's sort of where I think that they have bisected their hypotheses. Does it infect B cells? Ebola does not infect B cells. So B cells are one of the few cell types Ebola does not infect. I'm a hardcore mathematics fan, but you made me interested in biology. I love that. I love that. I think that other fields of study can appreciate biology because biology is just stories. And so I'm very happy to hear interdisciplinary interests in the kinds of stories that we tell here in biology. Okay, what does this toxic protein interact with? So that is something we will be answering in this journal club. Very exciting. So you be the scientist. Let's talk about this bisection of all possibilities. Let's say Ebola actually is making some sort of protein that interacts with T cells and causes them to die. So chat, how do we test that? What would we do to test this hypothesis? Go ahead and hydrate here while you all think of ways. And again, you don't have to have a biology background or a chemistry background or any science background at all. These answers can be answered very simply because I think that's what makes molecular biology so appealing, is that a lot of answers happen to be extremely simple. You just need the context to be able to implement it. I'm stupid, but I'm interested in your stuff. Disease bad. So it's been a while since I had to pull this out. I don't believe that the majority of people are stupid. I think that there's untrained. And so I highly discourage anybody from using that kind of language just because, you know, there's something psychological every time you do say that out loud about yourself, and it becomes almost a self-fulfilling prophecy. So one of my lines is like, don't call yourself stupid, because just... you're probably not, right? You're most likely, if you are, especially if you're in one of my streams and you are open to understanding some graduate level biology, there's no way I think that you are actually stupid. So please don't say that. Okay. Isolate the proteins and testing them in vitro. Certainly something that you can do. Prevent them from creating proteins and still see if they can do the thing. That's also a very good approach. It's an approach that they do take in the paper, but I actually cut it out for time. So Mylan, you are correct, but I didn't include that here because this PowerPoint is already 70 slides long. Isolating the proteins, that is a great option. Unfortunately, that is going to be difficult to do for biology reasons, but that's certainly something that I would have thought of as well. So that is not a a bad start. Knock out the proteins of interest in the virus. So that is certainly an option. However, that would require a hypothesis about which proteins. And so if you take that approach, you are torturing a PhD student with deleting a whole bunch of proteins when you haven't even proven that Ebola directly causes T-cell death. So that is an experiment that would be lovely to do eventually. But first, we have to prove if T-cells directly cause T-cell death. Okay, what else? What else do we have here? Synthesis of different proteins within the Ebola library, then exposing T-cells to their protein in a petri dish. Yes. Okay, so I guess I can talk about this now. So chat, that's a great idea. However, not all proteins fold very well when you purify them or make them yourself. So that is unfortunately just a physical limitation of something that they were toying with the idea of doing in this paper. So I'll say that yes, but maybe not for this particular virus. But again, we have to go in order. How do we tell if Ebola directly causes T-cell death? Directly. These are all great, and we're going to talk about those approaches, but we haven't even proven that this does this at all. Because if you think about it, if you do go through the trouble of making all of Ebola's proteins and then throwing them onto T cells, and that doesn't do anything, then you're kind of intellectually stuck, right? You've wasted a bunch of time for no payoff. So we have to go in order. It's a pretty straightforward experiment here. Ebola directly causing T-cell death. What else we got here? Wouldn't random synthesis of protein take an inefficiently long time? Exactly. It's certainly something that would be lovely to do, but we have to prove that we have to do that first. Just hopped on. What did I miss? Not much. Just starting with the intro here. Killed versus live strain plus T cells. See if there's an effect. I will take that. That is the closest answer to correct. In fact, that is probably better. I would say that's probably better than what they did live and dead strain to T cells. But yeah, so before we can do all this fancy protein work, we maybe just dump the protein on. Dump Ebola on, I mean. So the solution that the authors come out with is to grow T cells in a dish and then throw Ebola on and just see what happens, right? So these things about synthesizing protein, yeah, you could do it. There's no guarantee that it works, because when you make a protein, and it's not attached to the virus, it might not fold properly. So, if it doesn't work, you don't know why it doesn't work. So the better experiment is just to dump Ebola on and see what happens. Okay, so this is their data. So what can we conclude here? So let's warm up our brains and actually just look at some of this data. And so I have here a data key where I've defined some of the terms that you will need to interpret this data, to interpret this bar graph, right? So remember, Ebola does not infect T cells. They didn't prove that in this paper, but just, take our word for it. Ebola doesn't directly infect T cells. You dump the Ebola virus onto the T cell, and this is the data that you get. We are measuring cell death in panel A and panel C. I don't remember what the difference between... yes, okay. There are different populations of T cells, right? So annexin 5, this is a protein that binds to phosphatidylserine. So if you watch the Ebola video, you would know that Ebola coats itself with the host phosphatidylserine as a way to signal to macrophages that they should be eaten, essentially. Right, so phosphatidylserine, or PS, is a modified phospholipid that is a macropinocytosis activator. So it tells the macrophage, here's some cell garbage that needs to be cleaned up. And so we can measure the number of cells currently undergoing apoptosis, this programmed cell death, by measuring annexin levels, which is a proxy for phosphatidylserine. So this top panel, panel A, this is concerned with CD4 cells, or helper T cells. And so there's a certain pattern here. This panel in C, these are concerned with killer T cells. And so, this is the associated data. CD3/28 is specifically activated T cells, IMDCs, immature dendritic cells, MDCs, mature dendritic cells. So looking at this data, what can we tell about whether or not Ebola plus T cell equals death? What can we say? Does T-cell death have markers? They're not using a T-cell specific death marker, but since all these cells in the dish are in fact T-cells, a Nexin 5 will do just fine. All right, so what do we think? What trends can you observe here? If Ebola doesn't infect T-cells, what does this say? Yeah, Mylan says it does. Oh, by the way, Mylan is one of our newest members of Ethereum. If you are interested in psychology and anime, go ahead and check Mylan's channel out. Mature cells are more prone to death via Ebola. Let's take a look at that. So MDCs, it looks like there are increases. Whether or not they are more susceptible, statistically, they did not do that comparison, right? So you see the significance bars only in between the no virus and the yes virus conditions. So while subjectively we can say that, we don't actually know, officially what those statistics look like. And it's also not exactly a hypothesis that the authors bothered to pursue, but I suppose by looking at the magnitude, you might be able to conclude that. But yeah, so Mylan's got it right. All of the red bars, for the most part, all of the red bars? Yeah, pretty much all of the red bars are higher than their gray bar equivalent, right? So if you add Ebola to T cells, in the absence of other cell types, as you see CD4 alone and CD8 alone, you get a lot of apoptosis, right? And then you add the dendritic cells, and that seems to, just by eye, maybe it seems to sort of mitigate the effect, although it's hard to tell again because they didn't do the statistics on that. So that's pretty good evidence that T cells get killed by Ebola directly despite not being infected by them. Okay. Something that I learned from this paper, again, because I am an amateur immunologist, I'm an amateur virologist, etc., is that dumping Ebola onto the T cells actually also causes cell not differentiation, but replication of T cells. That is very unusual to me as someone who didn't know that this happens. So this is a separate experiment where they measured SFSE. So this is a cell dye. So you can imagine pumping these cells full of dye. And then if they proliferate, if they split, right, each cell gets half the amount of dye and then they grow and that dilutes the dye. And so to measure proliferation, you can stain these cells with this dye. And if a lot of cells are very faintly stained, you can say that they have replicated quite a bit. So something weird that happens is that when you add Ebola virus, not only do you get cell death, but you also get a lot of replication of those cells. And that's very bizarre to me. Very, very bizarre. That's such a fun method. It is. I actually didn't know that that's what CFSE was. I'm used to looking at apoptotic death signals, but this idea of just splitting the dye, very intuitive, not very hard to think about. But to me, the most elegant ideas are the ones that are as easy and fun as this. So it's kind of neat. Is the proliferation correlated to the killing method? I don't think so. I'm not entirely sure what that question means either. If you could elaborate on exactly what you're thinking, that might help. It's not actually that weird, apparently, as I dug into the literature a bit more. T-cell proliferation is a response to immune challenge. So if you have activated T-cells, if the T-cells sense that there's an infection going, well, it just makes sense that they would also proliferate, right? So this is something that is, I'm sure, for any immunologist out there, something quite basic, but it's also proof that I'm also still learning with you, which is why it's so fun to do these journal clubs, is that I have this moment of surprise of like, oh, okay, dumping Ebola onto T-cells actually increases the cell number as well as decreases the cell number, and these are different mechanisms, and that's pretty interesting. Okay, so there are going to be a lot of results in here. So all of these results are going to tie into one beautiful package, and I'm going to keep track of where the story is so you can see how careful these scientists are with their conclusions. So chat was jumping at the bit to make the protein, which, again, is something that the authors are going to do in some form, but to show you what it takes to publish a full story, we're going to keep track of all the painstaking detail the authors take care to do in order to tell a story that has I'm not going to say no flaws, but a story that is more unassailable. I'll call that. So the first thing that we learned is that Ebola kills T cells without the help from other cells and in absence of infection, right? So it's probably making something that kills T cells directly, which is what you were all suspecting. So you kind of already answered this question, chat. So Ebola virus is directly capable of killing T cells, but it doesn't require infection. How else might Ebola be killing off T cells, and how would you test that? I'm actually going to go ahead and continue on because your experimental ideas are more appropriate for this particular research question. So I agree that if Ebola is making some protein that's causing T cells to blow up, then we should try to make that protein and see if that protein is sufficient to blow up those T cells. Ebola surface proteins, exactly. Can you explain the root cause of cytokine storm in Ebola? So I'm not an immunology expert, but my understanding is that the absolute chaos and violence that Ebola causes a huge influx of inflammatory signalings in cytokine production. But there isn't like an anti-inflammatory balance because the immune system is thrown into such shock without mediation that storm proliferates and gets out of control, right? So again, in contrast to rabies, where it's very slow and very careful, Ebola is not slow and not careful. It jumps and explodes and is very violent. So yes, Ebola surface proteins. These are good candidates if you are imagining Ebola just touching the T cell and the T cell dying, going after a surface protein where Ebola does not need to be inside the cell and making all these other proteins. It's the most logical idea to identify a surface protein and see if that's the thing that kills T cells. Okay, so because, chat, your idea of making the protein is so good, that's essentially what they do, but they can't do it directly because when you make the glycoprotein in a dish, it doesn't fold exactly correctly. So instead, what the experimenters do is that they make something called a chimera, which is a very fun term. A chimeric virus is a virus that is engineered with genes from a separate virus. So we have this Ebola virus, and we want to see if their protein, their surface protein, GP, glycoprotein, we want to know if that's the protein responsible for killing T cells. So what we do is that we remove the Ebola, well, we don't remove, but we copy over the data, the blueprint, the DNA, or I guess in Ebola's case, the RNA, and we take that blueprint and we insert it into a different virus that does not exhibit the same characteristics as the virus that you care about. And so they chose para-influenza virus, so human para-influenza virus. So what they did is that they deleted these surface proteins of the human para-influenza virus and they replaced the surface protein with Ebola protein, right? So this is now a chimeric virus, one part para-influenza and one part Ebola. So the reason why the authors did this is so that the GP, the glycoprotein, would fold correctly because it needs to be attached to a membrane, a viral envelope, to be able to function properly. And by putting it on human para-influenza virus, we also then remove any of the associated variables on Ebola itself that could be contributing to the T-cell death. So we remove everything about Ebola, and then we take the thing that we care about and we put it into a separate virus. All right. So we kind of already answered this because chat got a little ahead of me in terms of where your scientific brains were. So why not just jump the GP onto T cells and see if it kills them? Why engineer a new virus? Protein folding. All right. So infection with chimeric para-influenza virus expressing Ebola GP is sufficient to induce apoptosis in peripheral mononuclear cells as well as isolated T cells. So peripheral mononuclear cells, this is like a messy sort of blood sample that contains a whole bunch of different cell types, T cells being one of them. So if we take a look on the left here in panel A, this is the mixed sample that contains T, B, NK, and monocytes. And we can see that in the green line, we have the chimeric virus, and that has a statistically significant difference in induction of apoptosis in these cells. And so there's a very good control here, and that's the unmodified human para-influenza virus, because again, we took the GP from Ebola and we put it into a new virus. Now that new virus has all sorts of different variables that are correlated with its existence, and so we have to control for that. And so doing the human para-influenza virus on its own is an excellent control here. And then when we purify the T cells, we actually see that the gap is a lot smaller between the mock, which is no virus, and the human para-influenza virus. So again, left is mixed sample, right is just T cells. So it looks like the human para-influenza virus really doesn't cause a whole bunch of cell death, not any more than not infecting with the virus at all. And then when you swap those surface proteins with Ebola's proteins, all of a sudden we get this significant increase in apoptosis. I know that Staph aureus makes some kind of extremely antigenic protein. Is this something similar? I don't know enough about Staph to really compare, and I don't know enough about Ebola to really compare. So while I did do a lot of research for Ebola, I'm nowhere near an expert. It's more like this is... Well, actually, I can answer that because the Ebola video answers that. The Ebola surface protein is not antigenic because the B cells don't make antibodies against it. The B cell would rather make antibodies against the soluble glycoprotein, which is not the correct protein to make the antibody against, which is another fascinating story. And again, if you haven't watched the Ebola video, I highly recommend that you do so. Very, very fun video. Could it be possible to separate the proteins via some sort of chromatography or gel electrophoresis? Yeah, testing the whole proteins, that's certainly something that you can try, but because they don't fold correctly, you have to do this chimeric virus approach. At least that's my understanding of why the authors did this. Okay. Literally, you dare use my own spells against me, Potter. It's true. Welcome, my constant Hadrian. Welcome, welcome. Welcome to the stream. Yeah, so... do we have concerns here? I actually don't remember why I put this here, so let's just continue. I think initially I was worried that the usage of the human para-influenza virus was going to be a little tricky, but I think now that I finished the paper, this has shown, like this experiment shows that the human para-influenza virus does not cause more apoptosis than not infecting with a virus at all. So I think this is actually really strong control. Okay, so we know Ebola glycoprotein is sufficient to kill T cells. What is the most logical next question to ask, chat? So I think maybe logical is maybe unfair. I should have worded this as, what is the question that you want to ask? So it might not necessarily be the most logical because that's subjective in some ways, but now that we know that GP is killing T cells directly, what's something that you would want to know next, chat? What's something that you would want to know? Because you can see that, like, there's not a lot of effort. I shouldn't say that, but there's not a lot of data that you need to generate to be able to prove that something kills something else. So what's the rest of these 70 slides going to be about? What does the glycoprotein do to kill T cells? What part of the GP kills the cells? Depends on the goal. What would your goal be, essentially? How? Yeah. What cellular pathways are being activated? Yes. Hey, Rico, welcome to the stream. Rico, a nurse VTuber, go check them out. What is the mechanism of action? Signal pathway. Exactly. So the next logical question that I would want to know, and what the authors want to know, is how the heck is this GP causing T cell death? And the line of investigation that they take, that's going to be specific, but that is the correct next step. Or I shouldn't say correct, but the step that the authors take. So, any hypotheses here? So, any models that you would want to test? So we have the research question that we identified, right? How does the Ebola glycoprotein kill T cells? That's our hypothesis. Not our hypothesis, that is our research question. So now we need a testable hypothesis. And then, of course, after we get a testable hypothesis, we need an experimental plan. So what are some ways that the GP can kill T cells directly? We've actually had some answers in the chat before, like maybe they're forming membrane pores that pop T cells, which is very cool, but I think is more of a thing that eukaryotes do to pathogens rather than viruses do to hosts. But that's not necessarily That's just from all the viruses that I have studied. I've never known one to do that, but it's not a bad idea. But what else could be going on here? What else could be going on? If you remove certain proteins from a T cell, will it still kill it? Interesting. So that would be an experimental approach, but not a hypothesis. Looking for a hypothesis here. Check what receptor the glycoprotein binds to. Certainly you can do something like a non-biased approach where you first try to see what the GP binds to. That's certainly a good idea. They actually don't have to do that in this paper because they have background knowledge. So I will say that A, you have the right answer given the information that I have provided, but they actually have a bit more information behind the scenes. Increases BCL2. I'm not entirely sure what that is. Capsase activation. That is a very specific hypothesis that naturally follows this idea of apoptosis, so that's not bad. But how would caspase be activated is something that they are going to be able to test here. So caspase is an enzyme that is a precursor to apoptosis, so certainly not a bad idea. Popping wouldn't incur apoptosis. Actually, you're right, icosahesion. That's absolutely correct. So if you do form a pore forming protein, that'll actually just pop the cell, which, I mean, depending on how it plays out, it could result in apoptosis if the cell gets stressed enough before it completely dies. Kind of interesting to think about, I think. Glycoprotein binding disrupts ion channels to trigger apoptosis. Yeah, so we are all getting around sort of the area by which the authors are thinking. And I believe someone else had actually said around like the correct answer. So checking what the protein binds to is what they are going to do. But because they have additional information than what you currently have, they have a target. So that is a good approach. So previous research has actually shown that Ebola glycoprotein binds dendritic cells through TLR4, so toll-like receptor 4. This is a membrane receptor that someone in chat had pointed out. The class of receptors this belongs to are the toll-likes. These are receptors, again, of the innate immune system. These are proteins that bind to pathogenic material. TLR4 specifically, I believe, binds to double-stranded RNA. So we also know that T cells also have TLR4, so there's a reasonable hypothesis that doesn't require checking every T cell protein, which you might have to do if you didn't have this background information. I don't want to make it seem like chat was wrong. You're absolutely correct that we are checking for what this protein binds to. It's just they had a little extra information, right? So this is a pretty good place for them to start. So let's talk about TLR4. So toll-like receptor 4, there are two pathways for this protein. There's one on the left and then one on the right. Not super important to know exactly how these work, but essentially they detect viral material, and there's a pathway that goes directly through the cytosol, so the body of the cell, and there's another one that causes the toll-like receptor to be internalized, and then that does its own signaling. And then if you watched my videos before, you would know that this eventually results in some kind of immune response. If you remove the protein that signals apoptosis, will the virus still kill T cells? They'll test something similar. That's a good thought. Keep that in mind. The compound that I mentioned before, mianserin, inhibits TLR4. Oh, that's very interesting. I wonder if they used that here, although I can't remember off the top of my head. So, how does one check if GP does bind to TLR4? Let's see. I've actually given this answer in my previous live streams, so let's see. If you are a biology student, then please refrain from answering this. Let's just, try to stimulate some creativity from the chat right now. Or if you know the answer from my previous live streams, then please refrain, at least for a little bit, right? So how do you check if glycoprotein binds to TLR4? If you're a complete non-biologist, I'm interested to hear what your intuition leads you to describing. How would one check for detection? Binding assay, electrophoresis. Those are certainly the correct answers, but I would like a little bit more of a mechanistic sort of story here, just to sort of appreciate how simple is not the right word, but how intuitive biology can be. Block TLR4 and see if it prevents death. That is a good experiment that eventually will be done. But in terms of physical interaction, does GP physically bind to TLR4? And that's certainly a different approach that the authors could have taken, right? You could have instead of checking for whether or not they physically interact, you could check for a result of them binding, right? So the story that the authors chose to do is this mechanistic binding first and then consequences, but you could also do consequence first. So Trojanet, that's not a bad answer. In fact, it's a very good one. It's just not the one that the authors are leading us towards, put it that way. X-ray crystallography is an excellent way to determine the structure by which two proteins bind and what it looks like when those proteins bind. Unfortunately, and there's no reason why you would know this, so don't feel bad, X-ray crystallography, as amazing as it is also very difficult to do. It requires a specialist, someone who is hired specifically to do X-ray crystallography in order to get that to work. So while that would potentially hypothetically work, it is not done at this level because it is so labor and cost intensive. Any other questions? Attach a quantifiable enzyme assay to the binding protein. That is very close to the correct answer. Very good. Put a bunch of markers like GLP on the TLR4 and see if it glows. So that's maybe something like FRET, which is if you add a fluorescent group to GP and a fluorescent group to TLR4, if those proteins do in fact bind, if those fluorophores like kiss, I like to say, if those fluorescent molecules that you attached, if they get close enough, one fluorescent molecule can transfer energy to the other fluorescence molecule, which can affect the light that is given off by their fluorescence. So that is certainly an idea. That's also a very difficult strategy to implement. It would work. Don't get me wrong. It would work. It's just very tricky to pull off. Would it be to do an ELISA? Something similar to an ELISA. All right. So what we have here, and so like ELISA would work as well. I don't want to say that that would be wrong. But what they instead do is something called a co-immunoprecipitation. So there are multiple different approaches to get the same answer, and chat's listed a bunch of answers that would also be correct. Here's what the authors do. This one is actually my preferred technique, just because this kind of binding happens within the cell. So there are advantages and disadvantages to using an ELISA. So for those of you who don't know, an ELISA is a very simple technique where you coat the bottom of a surface like glass or plastic with the protein that you want to bind to, and then you add in the protein that you think will bind. So we coat the bottom with TLR4, we add our purified glycoprotein, and then we add an antibody against the glycoprotein to see if anything stuck. So that antibody can be conjugated or attached to fluorescence molecules so you can measure light. That's not quite as good. It's a very quick way to get the answer, but because this is also my preference, because the immunoprecipitation happens within the cell, it replicates the environment of cell-like a little more reliably. Although now that I say that out loud, because this binding is predicted to happen outside of the cell, it's making me question whether or not co-immunoprecipitation really was the way to go. But in any case, it's fine. We'll see if that caveat affects our interpretation down the line. Because here's the thing that like a lot of young grad students get obsessed with, is that any flaw in a paper automatically makes the entire paper bad, which is not necessarily true, right? So here's the issue that we are coming together as a chat, as a stream, to sort of realize that the way that this experiment is done, the one that I have on stream, is that you ask a cell to make GP and TLR4 in its cell body. And then you break open the cells and you collect the juice, and then you see if those proteins bind. So I'll just explain this first, and then I'll explain why this is less than perfect and why chat has actually helped me to come to that realization, and also why, even though it's not perfect, why that's still probably fine. So you ask the cell to make both of these proteins by giving them the DNA for both of these proteins. You bust open the cells, so now the cell lysate, the juice that comes out of the cell, is floating full of glycoprotein, shown in purple here, as well as TLR4, which are these question mark receptors. and then you use TLR4 antibody as a fishing line. So here, let me get out of the way real quick. Here in orange, we have these beads, and these beads we attach this antibody to. This antibody is against TLR4, so it binds TLR4. And so this is like fishing for GP. So if TLR4 does bind GP, they should all bind together like this, right? If TLR4 does not bind glycoprotein, this TLR4 will be empty. So you can use this as a fishing line to try to purify glycoprotein by using TLR4 as a fishing line. Does that kind of make sense, chat? This is a really cool strategy, and I want to get a sense for whether or not it was understood. So does this make sense? I'll put a poll up. Use a centrifuge to sort particles by weight and see if you get any particles which is heavier than your inputs. So centrifugation can be a part of this story. If you have a particular hypothesis, so centrifugation is something that happens, and you know, when you process these samples, and if we have a hypothesis like this interaction happens in membranes, then centrifugation would actually allow us to just get the membrane part of the protein. So it's certainly an idea that the authors could have used, but I don't think that they did fractionation, although I could be wrong. I don't remember if they did fractionation. Wait, this gives me an idea for my project. We're trying to identify the TLR7 ligand in a system. Yeah, absolutely, man. Like looking at other papers and getting ideas from them, that's one of the best parts of science, right? So I like co-immunoprecipitation, but I actually don't know if it's appropriate for a membrane protein because you are asking them to make this intracellularly. And, you know, GPs presumably is interacting with TLR4 on the cell surface. So something like an ELISA isn't perfect because ELISAs require you to bust open the cells and purify the protein. Co-immunoprecipitation doesn't require as much purification because the fishing line purifies this for you. And then if we go back to Trojanet's suggestion about using fluorophorus FRET, now that would be a membrane specific test for binding, but it's a lot of effort. And so you got to make trade-offs with how important is this for this to be the best possible binding assay versus what do we consider to be good enough proof. It's an interesting question. So because this is not happening at the surface, it's a less than perfect assessment of whether or not this binding interaction is real. But that might not be a big deal because of compounding evidence. If we see that everything else points in the direction of TLR4 and GP actually binding, then we can kind of quote unquote forgive this being the less than perfect experiment to do. And that happens all the time in science, and it's not a reason to discount a paper. It's actually one of the skills that you learn as a graduate student towards the end of your time in graduate school. This idea that some flaws are acceptable is a very difficult thing to learn because you just don't know enough. But as you learn over time, you learn which flaws are deadly and which flaws are totally acceptable. No paper will be perfect, of course. I've always wondered with tests like this, would it have to be a very strong ligand for a result? Could this miss interactions that cause signaling but just don't stick together strong enough? Yeah, so that's a very good question, Joel. The answer is yes. So if the interaction is weak enough, you might not be able to purify enough GP for this to actually be captured. But this is sort of a problem no matter what technique you use, right? So any binding assay that you do, if the interaction is weak, you are just going to be less likely to identify it. In which case, something like an ELISA, where you can purify and concentrate and get the concentrations very high, that might be the better thing to do versus something like co-immunoprecipitation. So I hope chat can kind of see that like there are trade-offs. ELISA is more artificial because everything is purified and very clean. Co-immunoprecipitation is messier, but it is more cell-like. And so there are trade-offs in either system. And that's part of what makes doing the art of science so compelling, is that there's not one right answer. There are answers that are more artful than others. What do you mean when you say a ligand is weak or strong? Excellent question, Robert. So a strong ligand is so before I go on, a ligand is basically something that binds. So it can be protein, it can be an ion, it can be whatever, DNA, RNA. any molecule that binds another is called a ligand. And so a ligand is strong if it stays attached to that protein, essentially. And it is hard to peel off. That's simplifying a lot of binding kinetics that you can learn about in my Biochemistry 101 stream. But that's essentially simplified what that idea is, right? If it's sticky, then it's strong. If it's not sticky, then it's weak. Excellent questions. Okay, so we're doing this co-immunoprecipitation, or rather the authors are. We are asking the cell to make these proteins, and then we are using an antibody against TLR4 as a fishing line to try to see if we can purify glycoprotein from there. First, let's look at the controls. So a lysate is just the contents of the cell innards. We just bust open the cells with a bit of detergent and enzyme, and then I actually don't know if they use an enzyme, at least a little bit of detergent to pop the cells open, and then we use antibodies against proteins. And these antibodies are conjugated either with a fluorescent molecule or with an enzyme that can luminesce, right? So it can turn chemical molecules into chemical molecules that give off light. And so the darkness of these bands correlates with the amount of protein that was purified. So this is a Western blot. So if you haven't watched my short on Western blots, you can get very acquainted with how that works there. Essentially, to put it simply, we separated all of the proteins inside of the cell guts by size, and then we transferred them to a paper, and then that paper gets coated in antibodies against our targets. So let's look at the controls, because it's always important to look at controls. This is mock infected, so these are cells that were not infected with anything, and so we're testing for the presence of glycoprotein up here, GP, TLR4, and flag. So flag is an artificial piece of protein that researchers sometimes like to use when they're having problems purifying their protein. So you can kind of ignore the flag in this case. But if we don't infect these cells, and these are not T cells, these are some other cell type that's better at making proteins. Because again, we're not talking about T cells, we're just seeing these proteins bind, so it's not that important to be specific about the cell type that we're using. So in these cells, the mock infected cells, they're not making glycoprotein. Good, because we didn't ask them to, so this is clean. We didn't ask the cell to make TLR4, so that's clean, that's good. Flag, didn't ask it to make flag, so that's clean, so that's good. GAPDH, so this is a housekeeping gene. This is a protein that's always being made in the cell, pretty robust to changes. This is a proxy for how much total protein is in the cell. So yeah, there's protein in our mock cells, so that's great. When we ask these cells to make GP, glycoprotein, and TLR4, we see that GP has a band, we see that TLR4 has a band, no flag band, which is great. And then this band over here where we ask them to make the TLR4 that also has a flag tag. Again, this is a protein bit that is easier to, that helps with purification, essentially. Side question, are COVID and pregnancy tests a type of Western blot? Is it a related technique? I think that those are closer to ELISAs, which are, yes, those, I believe those are closer to ELISAs, which you can quickly look up. Or I'll just do a quick explanation again, no worries. You have a surface where you have a target protein, in our case GP or TLR4, and then you throw the other protein that you think will bind, and then an antibody against that that's conjugated to something that causes a visual change. I don't know about pregnancy tests, but something like an antigen test, I don't know about COVID specifically, but you can certainly do that, where you have an antibody against COVID on a surface, and then you flow the COVID on top, and then that captures the COVID, and then you have another antibody or another reagent that causes the color change. Yeah. Also applies to pregnancy tests. I see. Very cool. Very cool. All right. So yeah, so looking at the controls, it looks good because once you insert the genetic code in order for the cells to make the Ebola protein and TLR4, the cell makes it. Great. So their system is working as intended. So up here we have the actual data. Up here is the IP for TLR4, the immunoprecipitation. Up here is the flag. You can just ignore this because again, it's just there in case their proteins were hard to purify. So in the mock infected cells, we do not see TLR4, right? And then in the GP, when we give it the proteins, we do see that when we use TLR4 as a handle, that we can actually detect GP. So, yeah, so the IP, sorry. This is how this looks, right? So here's the bead. This is an antibody against TLR4, which is why this is labeled TLR4. And then the antibody that we are using is against glycoprotein. So there's a band here, which means we did fish glycoprotein out of the system by using TLR4 as a fishing rod. So this is pretty good evidence that GP, glycoprotein, binds TLR4. Is it perfect? No, they didn't do it on the cell surface, but at least in something that looks like a cell, with all the cell guts, it does bind. Any other controls that you would like to see here? What would be an important control here? Something that I actually didn't think of. So if you think of this control, you are better than me, because for me, I looked at this data, I was like, cool, we can move on. But there is a control that they did that I actually do think is pretty important, especially when we talk about viral proteins. So, and if chat can't come up with the control, that's fine. That means we're on the same level. But if someone gets it, that'll be a real embarrassment for me. And, you know, a streamer's embarrassment is content. So any other control? And I'll move on soon if chat is silent, but I'll give you a couple seconds or a minute or two to really rack your brain for one more control to determine whether or not this interaction is real and also meaningful. Well, sorry, so I guess here's my hint, right? Here's a hint. This interaction is real, but is it meaningful? There's a control that we can do to really ensure us, the reader, that this is a meaningful interaction. I'll maybe give you maybe like 30 seconds. Remove the interaction entirely. So what do you mean by that, Kahlo? What do you mean by remove the interaction? What does that mean? Co-IP MyD88? Maybe too niche? Oh, why MyD88, Wallace? Why choose that one in particular? Compare it with a similar TLR. Ooh, auto. So that is not what they did exactly, but that is in the spirit of what they did. So there is a control that you could do where you try to fish GP, not with TLR4, but maybe TLR3. Actually, they may have done it and I chose not to include it. I can't quite remember. It's not the data that I am about to show though. But I think they may have done that. So I think, Otto, you actually are correct. I just didn't choose to put that on the next slide. Is there anything we can attach to prove that it binds and activates? So activation will come next. That's not quite the control that they do here. And yes, that if we show that the sequelae of TLR4 binding are active, then that's good enough proof. But at the stage of just being like, is this interaction meaningful? We have to justify doing that experiment. I guess I would say Trojanet. So like, this is a real interaction, but we don't know yet if it is meaningful. Once we confirm it is meaningful, then we can go ahead and look at things like signaling pathways because we have more confidence that our work won't end up useless down the line. It's the adapter protein for TLR4. Oh, I see. MyD88 does come in this paper, but since I'm not like an immune expert, I actually didn't know that it is the... well, actually, I didn't really think about that. I knew it because we talk about MyD88 in this paper, but because I don't have a high-level understanding of this specific pathway, it didn't come to me as that's what you were getting at. A co-IP for MyD88, I don't really know if that would... Because then you've added another chain to the link, which might be just difficult to purify. I'm not saying it wouldn't work. I just imagined that it would be harder to get to work. Okay, so I'm going to go ahead and continue, and I'm going to say that Otto has the most correct answer. Instead of using a different TLR4, the authors go ahead and use a different Ebola protein. I actually think that using a different TLR is also a very good option. But in this case, they're using a different Ebola protein. So VP40, it's a different protein. And when we fish with TLR4, we actually find that it doesn't fish out VP40. So this is now, we can conclude, a decently meaningful interaction. Welcome, Peace Bone. Welcome to the stream. So we're kind of in the weeds right now. Right now we're determining whether or not the Ebola glycoprotein binds to TLR4, which is an innate immune system receptor that detects viral material. And so, so pulling down, so this is a test for what is called specificity. So is the TLR4 glycoprotein interaction specific? It looks like from the side of TLR4 that is true. So it binds glycoprotein, but it doesn't bind VP40. So this is a similar approach to using a different TLR4, because if you change the fishing rod and you still get glycoprotein, that's also an indication that this interaction is maybe not very specific, which would be a little bit of a headache when it comes to downstream stuff. So to address you, Trojanet, is there anything we can prove attached to prove that binds and activates? If we have a very messy protein, if GP binds to everything, that's going to make discussing what GP actually does very complex. Whereas if GP binding to TLR4 is specific, then we can start hunting for very specific hypotheses. Because anything that, like, say for example that GP binds also binds TLR3, well, that's a huge headache now, because then that would mean that anything to do with apoptosis, cell death, whatever, that could be the effect of TLR3 and not TLR4, which actually means, Otto, I think I might like your idea of changing the fishing rod better than changing the fish, right? So changing GP to VP40, that's changing the fish. Changing the TLR, I think I actually prefer that now that I'm talking about it in chat. And that's, I think, one of the greatest moments that I love doing journal clubs, because, I have X number of years in biology experience, but I actually do prepare these journal clubs very quickly, and so I don't often devote that much brain power to carefully critiquing everything. And like, the room that is there for people to come out and show their thinking brain is actually really great. There's also no guarantee that I would have thought to do TLR3 over glycoprotein as the swap. So I'm saying, like, I'm shielding my ego because I didn't think about that. But in reality, it's a great moment, because, like, could I have caught that? I don't actually know if I would have thought of that. Maybe I would have, but there's no guarantee. And the fact that there are people in the chat that can contribute to critiquing papers like this has always been one of my favorite features of doing these journal clubs. So that's very exciting. Hi, Peacebone. I've been here for 10 minutes and I'm lost. Yeah, it can be kind of tricky. I will do a bit of a summary when I get to the summary slides. So if you're feeling a bit lost because the molecular biology is kind of intense, there are like catch-up checkpoints that I have where we will talk through the story. Okay. So we know that Ebola protein, glycoprotein, this surface protein that coats the Ebola protein armor, it binds to this receptor TLR4. What else might we want to know? And it's something that chat has been screaming at us, at me. It is how. What is it doing? So we know that it binds to TLR4, but what is it actually for realsies doing? But before we do that, we just have to check one more thing. And so the co-IP, that's not really a perfect experiment, right? Because co-IPs, you ask the cell to make the two surface proteins inside the cell, and then you bust them open and see if they bind. That's not where they would normally bind, so co-IP is less than perfect. But to complement the co-IP, the authors do immunofluorescence. So using imaging to actually take pictures of the cell with glycoprotein attached, right? So here we have our T cells, CD4 helper T cells up here, DRICAT, these are immortalized T cells, and down here we have a non-T cell line, these are embryonic kidney cells, and then we throw on our chimeric para-influenza virus that's making the Ebola glycoprotein, and we actually see that there is red, which is the labeled Ebola glycoprotein around the cell proxy being blue here. So the blue is the nucleus, and the Ebola glycoprotein is binding around that nucleus. I would have preferred a cell membrane marker, but whatever. Like, what we can say is that the Ebola glycoprotein is binding to these cells. So because the co-IP isn't perfect, probably doing this experiment just to double check that this actually happens at the surface of cells, and it appears that that is true. Co-IP, that is co-immunoprecipitation. Go back a couple slides just to show how that works for anybody new. But we are using beads attached to antibodies against TLR4, which is the innate immune system receptor, and we are fishing for the Ebola glycoprotein to see if they bind. Are those good colors that they used? Red and blue, they're not red-green color blinding people, so I actually do prefer using blue and red. Also, these are pseudo-colored, so the authors can actually choose whatever color they want it to be, and choosing blue and red is ideal because, you know, just to be inclusive, you don't want to exclude people who are red-green colorblind from reading your data. What's the difference between T cells and the immortal T cells besides the existential dread? I mean, yeah, it's a good question. So immortalized T cells have been modified so that they can replicate on their own. So they are more robust in culture and are a common cell line to use when you're doing experiments. They are a little different than T cells, though, right? Because they have been modified, and the closer you get to an actual T cell, the better. Because anytime you use an immortalized cell line like HeLa, you are dealing with a bunch of changes that are not natural in a living cell. Okay. I don't actually know what modification is done to Jurkat cells, so I wouldn't be able to answer that further than that. I'm sorry about that, though. So how happy are we with this data? Are we okay with this? That we see the Ebola glycoprotein around the nucleus? Is this good enough evidence that GP binds TLR4? Very happy? I'm pretty happy with this, I think. This combined with the co-IP, this is pretty good evidence for me. All right. The authors do one more experiment. I actually really love this experiment because this is starting to get away from correlation and getting into causation, right? We know that they bind in the guts of the cell. We know that they bind on the surface, but do they really bind? Because here's the thing. This test, this is showing does GP bind, period. It has nothing to do with TLR4. The co-IP says that they bind inside the guts of the cell. This actually shows that they bind at the surface of the cell, but it's not TLR4 specific, right? We didn't do any TLR4 specific manipulations. So this experiment allows us to, I think, pretty cohesively say that this is actually happening, meaningful and real, that anti-TLR4 antibodies. So if we use an antibody to block the Ebola glycoprotein from binding TLR4, do we see an effect? So we have a bunch of T cells in a dish, we add the Ebola virus, and we add the anti-TLR4 antibody. And then we see, actually these are not T cells, sorry. If we're doing Ebola positive cells, these are a cell line that the Ebola virus can infect. So that is weird. Why do they do that? Now I'm confused. Give me a second to think about this. Interesting. So I thought they were using T cells. They might have been using T cells and then staining the Ebola for attachment. I think that's what they did, but I'm now no longer sure. The paper is linked in the description if anybody wants to check this real quick. It's pretty interesting. This does not account for a non-specific effect the protein has in the serum. Why the serum with anti-TLR4 antibodies and not the purified anti-TLR4? That's also a good question. No serum, negative serum, anti-TLR4 serum. And yeah, I'm not actually sure. Maybe it has to do with their culture system. So I'm not, like, I've never done this experiment for myself, so I'm not entirely sure why serum appears to be the default that they choose. I don't know, Wallace, if you have more insight into that, because that's not something that I have an answer to. It's a good question, though. In any case, using something with an anti-TLR4 antibody does inhibit some effect from Ebola onto whatever cell type they chose. Sorry, I did not look into this particular figure in as much detail as I probably should have. Okay, but here's our little bit of catch up. Ebola kills T cells without help from other cells and in absence of infection. This is the whole thing that is kind of sparked interest into the mechanism by which Ebola kills T cells. It's not infecting them, we know that from previous research, but it still kills them, and we know that it can kill them independent of other signals from other cell types. Ebola glycoprotein is sufficient to kill T cells, so when we move the Ebola glycoprotein to another virus that doesn't kill T cells, we see that just giving that harmless, quote-unquote harmless virus, the Ebola glycoprotein, causes them to kill T cells. So we know that the culprit, or one of the culprits, of Ebola's killing of T cells is this glycoprotein. We also just learned that Ebola glycoprotein binds to TLR4, which is an innate immune sensing protein. So that is where the story is so far, right? I'll go ahead and I'm going to give myself a second to just hydrate before we continue on in this paper. Maybe blocking TLR4 caused problems with the T cells. Yeah, maybe like serum is more friendly, like it's more conducive to happier cells, and that's why they use serum versus the purified antibody. It's actually a good question that I never thought to address. All right. Quick scan through the article. I still don't know why they use serums. Yeah, PLOS, despite the praise that I lop on PLOS for being open access and very forgiving with their copyright permission, they don't really explain a whole lot. And so reading a PLOS paper, reading a PLOS paper can be kind of hard. Whereas reading something like Nature, I think, is a lot easier. Okay, here's another interesting finding that they have, is that Ebola causes TLR4 levels to decrease, which points to this right pathway as being something that is activated, this internalization, right? So this is a graph showing the relative amount of TLR4, and when you add either Ebola or activated Ebola, you see a decrease in TLR4 levels, which, you know, maybe it's not doing this internalization, but it's certainly a possibility, right? So other things Ebola could be doing is inhibiting the production of TLR4. It could be doing something wacky that causes TLR4 to be destroyed, or because we're talking about TLR4 pathways, maybe it's activating this side of the pathway that causes the TLR4 to be sucked into the cell. Are these kind of studies have to be done in a BSL-4 lab, or BSL-3 is sufficient? Ebola is BSL-4, so it would be BSL-4. I believe in the paper it does say BSL-4 lab as well. Okay, so we have this point of data that points to this particular TLR4 pathway being activated. So then we might want to ask if this actually activates this, which is something that I've teased for like 1,000 years. Here we have a monster of a Western blot. Monster of a Western blot. Does the red have two peaks in the diagram? Yeah. This is another figure that I just kind of zoomed by and didn't really look at very carefully. Is this flow? Let me think. Yeah, I think this is flow cytometry. So this is... So let me explain this in a bit more detail. So this right here is the amount of TLR4, and on the y-axis is the population of cells. So we're taking cells that have been infected with Ebola, and then we are passing them through essentially a light machine, and that light machine essentially tells us how much of the TLR4 is on the surface. So when you have a mock infected cell, it clusters around this mean. So, you know, in this distribution, most cells have a decent chunk of TLR4, some have much more, some have a little less, right? Mock activated, that's also the same. And then when you infect with Ebola, more cells get dragged to the left here. So this is a population of cells that has less TLR4, and then the regular population. So it does have two peaks. this is the peak that has had the TLR4 removed, and then the normal peak that you see with the other cells. The diagram's confusing. It is confusing, and I should have thought about it a bit more, but I was playing too much Minecraft. I've been playing too much Minecraft. I started a VTheorem Minecraft server. It's private right now while we just play with each other and just have a good time. Not using it for content currently. But to my guilt, I have been playing Minecraft instead of double checking some of these graphs. Does glycoprotein inhibit TLR4, disabling production of cytokines and chemokines? Don't know yet. So we just see that the level is decreased. This pathway of TLR4 activation could be responsible. It might not be responsible. Who knows at this stage, but that is certainly a possibility. Does glycoprotein? I think it activates TLR4 and it kills the T cells. That is certainly a good hypothesis that I think the authors are rooting around for. Okay, so here's how we tell whether or not Ebola actually activates the TLR4 pathway, and it is via the Western blot. So again, I did a whole YouTube short on the Western blot if you want to see the Western blot in action. But essentially, what you have to know here is that we have a bunch of different TLR4 genes, or sorry, and here they're proteins, right? So this graph over here, right, this schematic that shows you how TLR4 works, the protein intermediates before it gets the signal to the nucleus, all of these proteins are represented, well, not all of them, but most of them are represented here, and we are testing for if those levels are elevated upon exposure to Ebola stuff. So this is certainly one way that we can test for whether or not the TLR4 pathway is activated. There are other ways, but the authors chose to use the Western blot. So here I have to take a quick poll, just in case like this is boring to people, because it's not like interesting to look at something like this visually. I prefer to look at fun fluorescent images, but I also acknowledge that a portion of my audience loves challenging themselves to learn how to interpret biological data, and Western blots are S-tier data. They are very important. If I were teaching a class, I would make every student learn what every single one of these lanes and every single one of these bands means, because as a biologist, you just have to do this. It is good for you. It just doesn't maybe make for the best content. So I will take a poll, because I don't want to be a judge of what is good and bad content. Do you want to go through this Western blot? And we won't go through the entire thing, but we will go through a good amount of it. What do we say here? Is there a safety gate for TLR4 that kills the cell if enough activation occurs? Almost like checking for faulty TLR4. So safety gate is a label that is a very human label, whereas, you know, a lot of biology purists will be, you know, that's personifying life, which, you know, you shouldn't do when you're talking about biology, because that can lead to assumptions that are faulty. I usually don't invoke that clause just because my whole channel is personifying biology, but in this case, the safety gate is going a bit far, because that implies a check. And I don't think, and your question will be answered for sure. But while there are checkpoints for stuff like DNA damage, I am unaware of a check-like mechanism for TLR4, is what I'll say there. To just the Western blot. Wow, overwhelming consensus that we want to look at these blots. That's actually crazy. Okay. Blots are the backbone of science. Let's do this. I'd like to learn. Teach us the Western blot. Let's freaking do it then. That's actually so... I wish my actual students were as excited as you all are. That's awesome. I love that. Okay, let's freaking do this then. Okay. So we first have to go through all of these labels, and I will eliminate a good chunk of these just because it gets repetitive, right? So you're not missing information. I'm just eliminating some of the redundance, but we will have to go through every single one of these lanes if we want to understand the best story here. So up here, these pluses and minuses, these are TLR4 inhibitors. So these are conditions where TLR4 is fully activated and conditions where they are not activated. And so you'll see how this plays into the story as we talk about the story. So we are checking essentially for the specificity of the response to TLR4 because biology is messy as heck. You know, a lot of different pathways have overlapping proteins, have overlapping structures and pathways. And so this direct inhibitor of TLR4 allows us to double check that what is actually being tested is in fact TLR4. Okay. These are TLR4 pathway intermediates with positive and negative association. So some of these increase during TLR4 activation, some of these decrease. We saw MyD here, which I believe Wallace had mentioned before. MyD is a positive activator of TLR4. So that's one example. And then the rest, some of these increase during infection, some of these decrease during infection. GAPDH, we saw our friendly housekeeper gene before. This is a proxy for total protein to make sure that we load in this experiment the same amount of total stuff so that we can actually compare. Okay, so we are going to eliminate this entire middle section because it gets repetitive. Let's just focus on phosphorylated TRAM1. So TRAM1 is one of these upstream. Is it upstream? Let me double check. TRAM1. Let's see, where is that on the thing? TRAM1 is in this right-hand side, right? So we have this hypothesis that Ebola is decreasing TLR4 through this right-hand side, so it makes sense that we're looking at TRAM. So TRAM is this guy over here, so it binds to TLR4 as it's being endocytosed into the cell, and when it's phosphorylated, it does something. I'm not a TLR4 expert, so I don't know what that something is, but essentially, if we are looking for pTRAM, we are looking for positive activation of viral infection. So the more pTRAM we have, TRAM1 that has had a phosphate chemical group added to it, the more that TLR4 is activated. Right? Okay. So first we look at our mock. Let's just look at mock right here. So mock, this is no infection, and we have yes TLR4 inhibitor and no TLR4 inhibitor, and we have no phospho-TRAM1, right? So this is not surprising. We don't add anything, and we get no TLR4 activation. Great. That's how that's supposed to work. When we add the inhibitor, nothing also happens. That's great. It'd be really weird if we added the TLR4 inhibitor and then we got TLR4 activation. So our controls look good. Now we have the second lane, LPS. So LPS is a bacterial lipopolysaccharide. So this is a sugar, a membrane sugar that bacteria make, and the immune system loves to freak out when it binds to this stuff. LPS is a highly, highly immunogenic activator of the immune system. So, you know, this is a sign that you have a bacterial infection. Pathogens love making this. The immune system finds this and it freaks out. So this is a known activator of TLR4. So in our case, this is a positive control for TLR4 activation. And what do we see, chat? In this band over here, this orange arrow. Does our system behave the way that we expect it to behave? What happens when we add LPS? Anybody in chat want to confirm that they understand how this is working? For you biologists, this might be easy, but for the non-biologist, this is a really good question to just check if you understand what we're looking at. This second band, we are comparing this second band right here to this first band right here. So mock, no inhibitor, to bacterial lipopolysaccharide here. And we're looking specifically at pTRAM, this one. This band compared to that band. What do we, first, let's describe what we see. So let's not interpret, but what is the actual look of the data? If you are a non-biologist, feel free to chime in. What does that look like? And feel free to full screen this data, by the way. It was hard to fit this on the screen with all the labels and stuff, but you might need to full screen the video to get a real good look at what this looks like. So chat, what's it look? By lane, do you mean horizontal or vertical? I mean vertical. So this here. They didn't align the thing very well either, which is kind of annoying, but this over here, this is the LPS column, and we want to compare it to this column where there's been no treatment. That blot is a part of the second lane, right? Yes, yes. It's unfortunately not aligned very well, but that is a part of the second lane. Correct, correct. When LPS is added without TLR4, pTRAM is detected. Yes, there's a mark on the blot for LPS where there is not a mark for the control. Exactly. So when we add LPS, we see a mark. That is what I am looking for. Yes. And so the conclusion, as Trojanus says, is LPS activates pTRAM, which implies pathway activation. Correct. So this is our positive control. This is what it looks like when we have successful TLR4 activation. We are comparing MOC, no inhibitor, to LPS plus no inhibitor, right? So this little smudge, that's our data there. So this is indication that pTRAM has been created, which implies that TLR4 has been activated. But that's actually not enough because, again, we are adding a TLR4 inhibitor to check if this is TLR4 specific, because LPS can cause pTRAM. Maybe pTRAM is activated by some other toll-like receptor, right? So we have to make sure that this is actually for real TLR4. And how do we tell that? How do we tell that, chat? Which lane, and you can use numbers 1, 2, 3, 4, which lane tells us if this is specific to TLR4? Start a poll. Which lane tells us that this is specific for TLR4? So lane 2 tells us that yes, when we add LPS, we have TLR4 activation. Lane 1 is what we are comparing to because that is no activation, which tells us that we do in fact have TLR4 specific activation of pTRAN. Lane 4. Great. Awesome. Awesome. Awesome job. Exactly. So when we add that TLR4 inhibitor, we remove the effect. So we are confident that this is a TLR4 mediated effect. Great job. Okay. So that's just our control. So you can see how much work goes into science like this, right? All of those lanes, that's just the control condition. Positive control is LPS, negative control is MOC. Okay. The next line here is poly-IC. So poly-IC is a viral activator of TLR3. TLR3, and it does not activate TLR4. So poly-IC is a double-stranded RNA analog, so it's double-stranded RNA, but it's been modified a little bit. And this, just like LPS, is the thing to add when you want to test for viral activation of the immune system. However, it does not act on TLR4. And since chat already has kind of figured out how to read this, I'll just compare the lanes that we need to compare myself. So comparing the poly-IC lanes, we see no band when we add poly-IC, and we see no band in the TLR4 lane. So we can actually say that this system, if we are using pTRAM as our system, pTRAM is specific for TLR4, not only because using TLR4, a drug against it causes it to shut down, but when we don't activate it, when we activate something else in the immune system like TLR3, we still don't get that response, right? So this is another great control just to double check, or in this case, triple check that TLR4 activation is in fact TLR4 activation, right? All of these are just the controls, but they are great controls to use. Over here in these lanes, we have our Ebola glycoprotein chimeric hybrids. Does anyone else see the very vague line in lane 7? Yes. So which one is lane 7? 4, 5, 6, 7. Yes. So now we're talking about this set of controls. So 7, I believe, is this one. And there is a very faint band there. That is for sure. And there's a good reason for that, because these two conditions, HPIV, EBO, GP, and this construct, these are both chimeric viruses that are expressing our Ebola glycoprotein. The difference between these is that this is the vanilla parainfluenza virus. Sorry, I'm about to burp. This is the vanilla, where my cursor go? This is the vanilla parainfluenza virus, and then you just add the glycoprotein. This is parainfluenza virus, but you swapped its existing membrane proteins with glycoprotein. So that's these two lanes here. And if you zoom in, it's very tough to see, but if you zoom in, you do see a faint band when you don't add the TLR4 inhibitor, and that band disappears when you add the TLR4 inhibitor. And using the same mentality that we've been chugging away at this Western blot for before, we conclude that this is specific activation of this system. Yeah. And so it's a little stronger, it looks like, when you do the membrane swap. Maybe there's less room or less competition, or maybe the membrane swap allows you to make more GP, but there's more of an effect here, at least by eye, which is notable, right? So this is, again, another sort of, well, this is not really a control, but this is like testing the hypothesis that it's GP that matters, right? So glycoprotein, this is not the Ebola virus, but it is a virus making the Ebola protein, and we do get TLR4 activation, which disappears when you inhibit TLR4. So that's pretty good. Does the strength/visibility of the band matter? It does, but because they didn't plot the data, we can only say by eye, followed by an asterisk, right? The darker the band is, the more that protein is activated, but unless we have the number, we're just kind of guessing. Because you also have to divide by the total protein, which is this GAP-DH band here. So, you know, make any assumptions about the relative density, the relative darkness at your own peril, right? Because eventually all of these numbers have to be divided by the darkness of this band here, which I don't think they did in the paper. Okay. And on the right here, all the way on the right, we have our vanilla virus, para-influenza virus, and our vanilla Ebola virus. And both Ebola virus and para-influenza virus actually do activate TLR4. And this, chat, why do you think this is a headache for me? Why am I a little upset at this finding that both of these viruses are activating TLR4? Why is this super annoying? What about that result is a headache, right? And it's the strongest bands, right? The vanilla para-influenza virus, there's a band here. Vanilla Ebola virus, there's a band here. They both go away when you add the TLR4 inhibitor, so it's specific to TLR4. So why does this make me cringe? Just a little bit. Anybody know why I would be shooketh by this result? It removes the entire purpose of using that virus as a vessel, because it suggests that paraflu kills T cells. So, Jareer, that's something that we actually covered before. It's okay if either you forgot or you weren't there for that part of the stream, but we know that para-influenza virus does not cause apoptosis in T cells. That much is true, and that's actually a good point. That's why I'm not like super upset at this data, but I am bummed out. Because you can't be sure if your viral chassis was messing with your data. Yeah. Now it doesn't determine if GP is the one, it occurs with both. Yes, exactly. So, you know, we did this Ebola thing where we made chimeric viruses because we wanted to remove Ebola virus itself as a variable that we had to consider, but our viral chassis also causes TLR4 activation. That is super duper annoying. I'm not upset entirely, because of what Jareer had said and what Jareer had brought up. We know that the virus doesn't kill the T cells with apoptosis, so it's not like it was useless to do in certain ways, but this finding that the para-influenza virus does also activate TLR4, that it's kind of annoying, and it kind of makes the waters a bit muddier. But again, when we aggregate all of the evidence, the co-IP, the fluorescence data, et cetera, and this activation data and the inhibitor data, like the picture overall still looks good. It's just a little smudged now because the viral chassis alone can activate TLR4. It's just a little annoying. Hopefully some of the other rows below indicate some differences. I think the PTRAM one was the most straightforward one, although let's take a look if we have... Yeah, I don't think, if we're just looking at here, because this is the lane that really upset me, it looks like IRAK4 shows the same behavior. Yeah. I don't think the other lanes will have cleared that up, to be honest. Okay. So it's a little bit of a headache, right? Confounding variable man. but it's not that big of a headache. It's okay, I think, because we have other lines of evidence to supplement what we currently know. A little different in phospho-PYK2. Let's see. No, not really. The behavior is the same, right? Like we still get those bands. We still get that positive activation. This is still here. Essentially, these are all littered with TLR activation, and that is pretty upsetting. We want to see this lane be different than this lane, right? We want to see this lane 1 be different from lane 3, and it looks like in every condition tested, it's about the same. Bummer. Confounding variable, man, my enemy. Yeah, for those of you who maybe jumped in after I introduced Myelin, the latest member of Ethereum, a psychologist, and I've had many a fun discussion behind the scene and also on stream because we were collaborators on my channel about how annoying statistics are and confounds. If you want the final boss of confounding variables, it's social sciences. So while this is a headache for me, you know, I'm sure Myelin deals with confounding variable man much more frequently than I do. Okay. And also, again, this idea that like having anti-TLR4 like, you know, there are other lines of evidence that show that the binding is probably specific. It's just a little muddied because of the chassis thing. Okay, so here's the results. Catch up, I guess. Oh, you know what? That's so crazy. I didn't even realize. YouTube is filtering my chat. Whoa. I did not even realize that. Icosahedron. And for anybody who I didn't respond to, so by default, YouTube chat is set to top chat, which doesn't include every message, which is kind of crazy. And I just realized that because I have OBS on, and OBS actually showed me a message that didn't appear in this other tab that I'm keeping track of. So that's really bizarre. So icosahedron, what would have been a better approach though? Stripping the virus of whether or not of whatever activates TLR4. I think that would have been cool to do. Yeah, like a domain swap for Ebola, because there's a chance if you delete GP from Ebola, it just doesn't work. You just brick the whole virus. But one potential thing to do would have been to swap GP for something on para-influenza virus. That would have been cool. Might have been difficult because I don't know how easily engineerable Ebola is, and you want to do as little engineering in BSL-4 agents as you physically can, right? Para-influenza, not as big of a deal. Any genetic engineering done in Ebola has to be done with very, very good reason, just because you want to limit the risk of like the world ending, right? So yeah, I'm sorry if I missed your message due to YouTube filtering the chat. That's so crazy. Okay, so results tracker. Oh, and no worries. Go do your groceries shopping. So here's what we learned so far. Ebola kills T cells without the help of other cells in an absence of infection. It can kill T cells directly, and it's mediated through the GP protein. We think the GP protein is activating TLR4. We know it binds to TLR4. It's probably binding TLR4 like with high certainty, but not 100% certainty. And we know that once it binds to TLR4, there is a productive activation of the TLR4 pathway. Genetically modified Ebola indeed sounds like the doomsday gizmo of the end boss. Yeah, exactly. So like, I think icosahedron's idea of like stripping Ebola of its viral protein, I think that's a good idea. I wonder, it's my hypothesis purely. I wonder if they didn't because of BSL-4 restrictions. I worked in a BSL-2 lab, so I worked with real baby stuff. So I wonder if that's the kind of thing that they have to be aware of when you get up to that high level. I just started hearing Dark Souls music, true. I used to want to be a prion researcher. I actually wrote a grant when I was an undergrad. It was the first grant that I seriously sat down to write. I got rejected, of course, but I really wanted to study prions. And having worked in a BSL-2 lab, I can guarantee you that would have been the wrong life choice for me. I am too messy of a researcher to be trusted with BSL-4 reagents. Welcome, Spepic. Welcome. Okay. So we know that there is activation of TLR4 pathway, and some commenters have mentioned that activation of TLR4 pathway can result in cell death. However, the exact details of how that happens is very, very messy, right? So, we have this pathway, it's nice and neat, and then we have vaguely down here pro-inflammatory cytokines, type 1 interferon cytokines / chemokines. So in this simplification of genetic activation, we have like a gazillion genes, right? So yeah, great job catching up, Spepix. So that's exactly where we're at right now. We need to find out how TLR4 causes actual cell death. And so there are like a gazillion things that are simplified in this part of the schematic, and the researchers want to know what things are being activated when Ebola is present. So that is interesting to note. So here's what the researchers are going to do. They're going to do deep sequencing of RNA of infected T cells, infected in quotes, right? Ebola doesn't actually infect T cells. It doesn't go inside and replicate, but they dump the Ebola virus onto these T cells. And then when we deep sequence the RNA, take out all the RNA and then read them, essentially, we can then see which genes are turned on after Ebola infection. And we can actually have a more specific answer if we compare to T cells that have been activated with bacterial LPS, that bacterial lipopolysaccharide that we had mentioned during the Western blot, this bacterial cell wall sugar that the immune system loves to freak out over. So this allows us to narrow down a set of interesting target genes that are specific for an Ebola reaction. So that would be very interesting to know. So Ebola has a bias towards activating necrosis instead of apoptosis genes. So you do this deep sequencing, you look at all the RNA that is changed with Ebola infection compared to no infection and compared to bacterial cell wall infection, and you see that there are differences in these broad categories of genes, right? So here are the categories they tested for: apoptosis genes, necrosis genes, toll-like receptor genes. So this is immune activation, and this is cell death. These are all like related to one another in a huge, horrible mess of genetic pathways that genetics nerds love thinking about. But for someone who is much more of a classic bread and butter molecules kind of guy like me, this kind of stuff is always a huge headache for me. But what we can see here is that in Ebola exposed T cells, after 4 days, you get, apoptosis genes and you get necrosis genes. And on day 1, you get a lot of necrosis genes being expressed. And when you just use the bacteria cell wall, you get fewer of those genes being expressed. So you can kind of peruse the numbers if you want to rewatch the VOD and pause and take a look at some of the interesting numbers that come out of this. But the idea here is that exposure to Ebola looks pretty similar to bacterial lipopolysaccharide, but not quite the same. And those subtle differences are going to be what's interesting about Ebola, or what's specific to Ebola here. What is the difference between necrosis and apoptosis? That is an awesome question that we are, I think, just about to answer. Yeah. So, Robert, you have predicted the relevant thing for the audience to know here. So, apoptosis is the thing that I talk overwhelmingly about. I talked about necrosis a little bit in the tuberculosis video, but apoptosis is a cell going out on its own terms. It is programmed cell death. It is a humane way for the cell to give up its own life in order to better the body. If you are a cell and you're going to die, you are going to choose apoptosis over essentially any other pathway. It is the cleanest way to go. So what happens in apoptosis? You have a cell, you start blebbing at the membrane, and then the cell essentially disintegrates into smaller blebs. And during this disintegration, from the Ebola video, you get flipping out of phosphotidylserine, right? So this is a way for the immune system to easily clean up after these dead bodies so that they don't continue to hang out and cause immune activation, right? So this is a convenient cell death that allows the immune system to clean it up very quickly and repair whatever damage needs to be repaired. I liken apoptosis to the masculine urge to bleed out in a snow-covered park, right? If you want to choose a romantic death, you choose apoptosis here. Is bleb the official science term for this? Absolutely, it is blebbing. They are called blebs. Necrosis, on the other hand, is like Breaking Bad, where you are unceremoniously shot in a dark alleyway. This is not the way you want to go. So instead of peacefully breaking up and disintegrating this humane death, you are essentially just popping and dying. So small blebs form, the nucleus starts to break down, the blebs grow and freak out, and it just pops. There's no time, there's no system to do this in a way that is convenient. These cell ruptures, and importantly, this rupturing is actually really important because here the destruction is self-contained, right? Like there's not a ton of material that actually escapes out because the cell's death is confined and regulated in these tiny blebs. Necrosis, you pop the cell. That includes all the guts in the cell. And the guts, if they're free-floating, can activate the immune system, can cause inflammation, can cause damage. This is the type of damage that is present in things like necrotizing tissue, right? So this black, ugly, damaged tissue that you get when you have particularly severe surface infections that kills and eats away at your flesh. That's necrosis, right? That's bad news. That's damage. That's dangerous. Anytime I even read the word necrosis, I actually do feel a little bit of terror because apoptosis, while tragic for the cell in question, is what the cell was programmed to do. Necrosis is again, unceremoniously getting shot in the back alleyway. Not good for the cell. If I add detergent to a cell, I cause necrosis. Detergent might be too fast for necrosis. Detergent might just be instant, like, dissolution. It's a good question, though, whether or not if you add a light amount of detergent, if necrosis is technically activated. And these are pathways too, right? So there have to be time for this to be set up, so it's not instant death like detergent would be. Apoptosis is like cell, whereas necrosis is like a cell, yeah, exactly. In terms of like people complaining about YouTube censorship, I've been demonetized for less, so let's just put it that way. Okay, so all of these genes, you know, they're just numbers to you. Some go up, some go down. And here is why I will never be a geneticist. These horrible networks of genes, dude, like TLR4 activation, and then you get all of these nodes that connect to all these other genes, and they all regulate each other, and some cause these to go up and some cause these to go down, but then these genes work together with these genes. Some go up, some go down. It's a whole nightmare mess, tangled web of horribleness. And that's just the daily life of a geneticist. So that's just what they do. It's not the science that I prefer. But what's important to know is that there are networks of inflammatory activation. This is also why infection is so hard to study, because the cell is hard to study. It's also why cancer is hard to study, because cancer not only includes potential genes from your cell cycle, from your DNA repair cycle, but also includes all of your inflammatory genes and your immune system genes, and you know, I'll talk a lot about it in my stjudes glioblastoma stream, but like cancer, infection, immune system stuff, it's all a terrible, horrible, tangled mess of interacting proteins. So is the ungraceful exit of necrosis like an emergency blowout stop for a cell that's going way, way wrong and has no time to do things gracefully, or no method to do things gracefully? Exactly. It is not preferred. It is not the preferred way to go. They are worse to clean up as well. You want to go by apoptosis. Okay, so this one in particular is important. TNF, tumor necrosis factor. So you can tell already that this is a gene originally identified to be important in cancer. And again, because cancer is so complicated, it involves interacting mechanisms from pretty much every system in your body, right? That includes immune, replication, metabolism, you know, a zillion things contribute to cancer, and then a fraction of that zillion things are the immune system genes. So TNF is a particularly important regulator here. So, to some, Ebola kills T cells. No help from others. No infection needed. The protein is enough to kill the T cells. It binds to TLR4, which is an immune sensing protein. That binding does, in fact, cause activation. And that activation is correlated. We'll say correlated because I don't remember if they did a TLR4 blocker. But that activation is correlated to an increase in apoptosis and necrosis genes with a bias towards necrosis, including this central immune signaler TNF alpha, tumor necrosis factor. Sorry, how can you look at this graph and say this is more important than any of the other ones? Or was that graph just an example of many different ways they exist? So that's a good question, and it's a question that I think that only an immunologist can answer. I happen to believe that this is important because TNF itself is very important. Like if you want big name genes that most people would recognize, you know, TNF is recognizable, STAT1 is very recognizable. I'm going to show my whole ass by like not naming every one of these significant ones, but in terms of like a non-immune guy like myself, TNF is something that even I have heard of, even in grad school, right? So something that like you don't have to specialize in immunology to know how important TNF alpha is. And also probably previous literature gives them a convenient model, because like what if the actual important thing was I don't know, FSTL1, I have no idea what this is. If this was actually the important thing, but you know not a lot about it, you might not be able to do anything about it. So picking something that has good name brand recognition is not a bad way to try to hedge for success, I guess you could say. So is GP just using TLR4 to get in so that it can cause big trouble? So it can't get in, right? So Ebola doesn't infect T cells. So it's using TLR4 just to blow the T cell up, to prevent the T cell from stopping Ebola's rampage. Right, so it doesn't follow from just the info shown in this graph then. Yeah, I'd say so. It's a little misleading the way that I'd set it up. All right. Okay, so here we have testing for a TNF alpha reaction. So this is testing for the amount of TNF alpha, mock, so no infection, Ebola virus, and then staphylococcal enterotoxin B. So this is the positive control. And we do see that when you add Ebola virus to cell culture that you do get an increase in TNF alpha. So this is correlated with TNF alpha being an important part of the story. So not going to redo the whole list, but this is one of those things that is upregulated in this correlative apoptosis necrosis gene mess, is this TNF alpha, and confirmed. And then so the authors confirm that this does actually happen. So, you know, gene expression is nice and fun, but protein is where it's actually at. And when they test the protein, this does appear to be true. We also know that if you dump TNF alpha onto these cells, that you do get more dead cells. So TNF alpha is sufficient to cause cell death. I don't exactly know how they picked a concentration, because of course you know that anything can be a poison in high enough concentration. So this is kind of, to me, a little bit unnecessary, but sure. So we can add that technically. TNF alpha exposure is sufficient to cause T cell death. This is a bit of a tenuous connection, but I get why they did it. Just to show that TNF alpha can kill T cells. Although again, I'm skeptical of why, you know, like how good that connection is, but it might not need to be that good depending on what they have going on elsewhere in the paper. And that is this. So I hope you understand why like this is a tenuous connection, right? If you add TNF alpha, it kills T cells, but like how much? Again, you add any amount of immune signaling protein and eventually the cell is going to die, just because that's how the immune system works. But this is the actual part that makes that data okay, I think. If you block TNF alpha with an antagonist, that actually stops Ebola from killing the T cell. Now that is gold. That is gold. This is an actually good example of like how one imperfect experiment that is like, I don't really love it, it can be kind of whatever, that like kind of so-so experiment can set up a much more beautiful experiment, which is this one. So blocking TNF alpha, it doesn't improve the overall cell death, right? So like if you have this mock infected, no Ebola infection, you add that Ebola infection, you get death. Obviously, that's what we've been talking about this entire time. When you do the same thing, but you stop TNF alpha from working by using an antagonist, T cells no longer die. Now everything, all of the previous lines that have been very tenuous, for me personally, all of those beautiful pieces of data have clicked into place with this 8th point, right? So TNF alpha, sure, it can cause T cell death, but is that happening in Ebola? Who knows? When you block TNF signaling, you prevent Ebola from killing T cells. That's super interesting. We also know, if we're talking about whether or not those necrosis pathways matter, if you add necrosis inhibitors like X5GA1400W, I have no idea what these are, but sure, if you tell me they're necrosis inhibitors, I'll believe you. They also stop Ebola killing, so that also gets to be added to the stories. So TNF alpha is involved in necrosis. Blocking that necrosis also prevents Ebola from killing T cells. So these are all circumstantial, building upon one another, but it is a compelling story. So here's the big picture here. Ebola protein binds to TLR4 through its glycoprotein, which activates TNF alpha. Probably, right? Like, we know that TLR4 is correlated to TNF alpha, but because we do not see a TLR4 inhibition experiment, I don't really know if they did one. I think they might have, but maybe I skipped over it by accident. So I'm not going to say that I didn't do it. I'm just going to say that I didn't show it. But if you do this experiment where you block where you block TLR4 and you see if that stops TNF alpha from being activated, then you can make this entire connection, and then we're all chill, right? But this is essentially how the story goes. Ebola binds to this protein that causes an activation of stuff in the cell, which eventually causes TNF alpha, and then TNF alpha can result in necrosis plus apoptosis. It's probably not the only gene involved in necrosis plus apoptosis, but we do know that if you stop TNF alpha from working, that you essentially preserve the T cells entirely. So while it's not a guarantee that TNF alpha is the only apoptosis necrosis gene that matters, it is pretty good evidence that this is a worthwhile target to go after when it comes to designing Ebola treatments. So TLR4 blockade might be useful for treating Ebola. Maybe not TLR4. I mean, maybe yes, maybe not. This has also revealed TNF alpha to be a very good target as well, because I didn't see in the paper this experiment with TLR4 antibody instead, and I don't know why that is. Again, I think that PLOS papers have a pretty hard time telling a complete story, and I just have to like come up with justifications myself. But you do bring up an excellent point that like this certainly points to TLR4 being a good target for Ebola treatment, as well as TNF alpha being a good target for treatment. So you are all right. That is definitely where, you know, minds are going towards. Could blocking some of these increase cancer risk because cells stop dying? Depends, right? So there aren't just one or two cell death pathways. There are a whole bunch, and the cell death pathways, like apoptosis, has so many things that feed into apoptosis. You can be triggered by the immune system. You can be triggered by DNA repair. It can be triggered by failure to split properly. So it is probably worth it. And the other thing is that you have to consider that in medicine, no medicines are perfect, right? So if you are dying from Ebola infection, I am certainly not going to complain about taking a drug that slightly increases my risk for cancer to stop myself from bleeding out from the inside, right? So the answer is, I think probably not, unless TLR4 is something that the NK cell needs to kill cancer cells, right? Because that could be an issue, but I don't know enough about NK cells to say that confidently. Weird. PLOS doesn't have a word limit, if I recall correctly. Yeah, I don't know. Like every PLOS paper I've had on stream, I've enjoyed the results, but the making of the PowerPoint has been kind of painful because I so often have to come up with my own story. Why do cells conserve these necrosis genes if they can trigger accidental death? Necrosis is the death, basically. I'm not entirely sure, because that's a pretty difficult question to answer. My guess would be it's better to die in any fashion than it is to allow a virus to co-opt your body for viral replication, would be my guess as to how that evolved. Yeah. So why does TLR4 have the capacity to induce cell death at all? It's really messy. So like, you know, nature is not exactly a clean programmer, right? A clean tinkerer, a clean engineer. A lot of these things just happen to be good enough to allow us to continue living to this day. And so having a lot of immune stuff hooked up to apoptosis is probably a okay solution to ensure that your cells cannot be continuously parasitized by pathogens. So I think while it is while it is exploitable, it's not without justification, I think. It will increase risk of cancer because the patient will survive. Classic. That is true. That is very, very true. Very, very true. Okay. All right. So here's a bonus figure. Again, I find plot stories to be so crazy because sometimes it's just all over the place. But here's a bonus thing that they added right at the end that has nothing to do with T cells, right? Like this is, again, this would never happen in a Nature paper. Nature paper is a very tight, very well told stories, and here this paper about T cells, all of a sudden we're talking about macrophages for some reason. Okay, so Ebola forces the THP1 precursor cell to differentiate into macrophages, its preferred victim. Unfortunately, I haven't seen a good negative control here, which is kind of unfortunate, but this is kind of a cool note to end the paper on. So if you watch the Ebola video, and if you haven't, please do. It's good. I worked really hard on it. You would know that Ebola's favorite cell to parasitize, favorite cells are macrophages and dendritic cells, and also monocytes, but I thought that 3 main characters would be kind of much for the Ebola video, so I just ignored them. But just know that they're also part of the story. But macrophages are certainly one of their preferred foods to snack on, right? And so what this experiment is showing, CD14 and CD11b, these are markers for macrophages, and we actually see that when you add Ebola virus, you get differentiation into macrophages. So you shift from monocyte cell fate away from the other fates that THP1 cells can differentiate into, and you force them to become macrophages, which provides more bodies for the Ebola virus to replicate in, which is just insane. Weirdly enough, right? Like, I'm only saying that comparing orange bars to mock bars, right? You do get this differentiation. You do get this forceful differentiation in this top figure. And it does appear to be like TLR4 dependent because that's what this is. So if you inhibit the TLR4 response, that can decrease the amount of differentiation into macrophages. Although, again, that's, and I think, not the best negative control, but that's fine. LPS does it too. So bacterial liposaccharide, lipopolysaccharide, that causes macrophage differentiation, like a huge amount of macrophage differentiation, which I guess kind of makes sense, right? Because you are saying to the immune system that there's bacteria around, and so that would probably recruit macrophages to do their macrophage thing. But it's interesting that Ebola can do the same. And when you swap the para-influenza virus with the Ebola virus, when you swap their proteins, the para-influenza virus actually becomes better at making macrophages with this macrophage marker than the Ebola virus is, which is actually kind of wild, and I have no good explanation for that. And down here we have viral infection, I believe. I don't remember exactly what this figure is, but yeah. This is the last figure. I slapped it on in the last minute, and I don't remember why I have this here now. Ebola GFP %. This implies that this is infection with Ebola. But then we have mock here, and I don't really know how you can have a mock non-infection. So that's weird. So let's just ignore this figure down here, just end the story up here. Ebola forcing these THP1 precursors to become macrophages for further predation is a pretty cool mechanism that I kind of wish that I had put in the main video. It almost feels as if this data point could be worthy of being published in its own paper, sort of a look at we found when we looked for something else. Exactly. What's the DOI? The paper is linked in the description. Could it be a case where the Ebola was as good as the influenza because it died out too fast? Maybe. I guess I wouldn't know. I mean, that is true. There's no control for like death here that I have on screen. I don't know if they did one, but that is true. Maybe this is artificially low because there are just fewer surviving macrophages because they have been gimped by the by the Ebola virus. Yeah, so that's a good point. That's a good confounding variable, man, coming back in to heckle us once more. But what is true is that this is greater than mock, and that's the important comparison to make. All right, that is it. Are there any questions? Let's go ahead and end on my plug for my glioblastoma stream. this one. So we got Stream Elements bot in the chat if you want to ask questions about the glioblastoma stream, but I will move into the just chatting portion of my stream now. Just kind of hang out with all of you for a little bit. Yeah, so any other questions about Ebola while I take a little break to hydrate? This was a really fun journal club to put together, and it was really fun doing research for Ebola. But I will say that, like, I am aware in my, like, very privileged position that Ebola is not an actual threat to my existence currently. And so there is this tendency to be like, oh, you know, this horrifying disease that only affects the other side of the world will never affect me. But um, it is important to sort of maintain a sense of vigilance when it comes to watching out for our neighbor, right? In terms of political boundaries, viruses don't care. I mean, we all saw that with COVID, right? One country helping another preventing the spread of viruses in their country ultimately will benefit every country, right? Not just the country affected by whatever is happening, right? And I remember back during the Ebola scare that there was a lot of humor that was happening, which, I mean, you know, people cope however they cope, right? But as like an edgy kid, I also thought it was funny. As an adult, I absolutely do not think it is funny. As an adult, I'm like, oh, okay. Yeah, reading the stories of how Ebola has affected those countries has been very harrowing. And while I have been picking like easy targets for first world countries to be like, oh, you know, rabies, scary virus, you know, doesn't really affect me at all. Some of the viruses that I'm about to cover, some of the pathogens that I'm about to cover, can very well threaten first world countries as well, especially if the response is, you know, if the response is bungled, if the ball is dropped, some of those pathogens. I am making these videos not out of a sense of like joy in terms of like, oh, you know, personally I'm not terrified, but you know, mechanisms are interesting. These next few ones are motivated by my own anxiety. I'm going to try to not let that like bleed out into the script, but just so that we are all clear on why I do the content that I do it primarily because I think it's interesting, but because the world is the world, because the world is the way that the world is, there is going to be a little bit more of sort of a plea for public health, put it that way. What a good Christmas present, Ebola. You are welcome. Seeing that MRI makes me really think of a spoopy brain infection called Balamuthia. Oh, interesting. I might have to look into that. Any sneak peeks about what's coming up? Well, there's this glioblastoma stream that I'll be doing on 1/11, and the next pathogen will be a virus, and it is a virus that is, I'll say this as a teaser, it is one of humanity's all-time worst pathogens historically, and if we are not careful, that might not even be historically anymore, right? We might be living that terror. So watch out for that. Ebola was once at the border of my country. When I was younger, I did not care because the name was kind of funny. Yeah, when I was a kid, it was hilarious, right? You know, oh, there's people on the other side of the world, haha. You know, like it's funny, like because it wasn't on our shore. But you know, having a global perspective is important because if you've ever played Pandemic or Plague Inc. And also it's just nice to have empathy for people who don't share your exact same living situation. But if it takes sort of reminding people that we are a global species and that viruses don't care about political borders, then it is worth doing so. Measles, smallpox, these are good, very good guesses. I will say with measles and smallpox, historically they have been easier to combat. This one's hard to combat, and that's all I'll be teasing. TB, I covered TB already. It was actually the first video I've ever done, if you didn't know. So if you go to my video tab, the first video I ever did in the style of Tirzu was tuberculosis. I still think that video is pretty good. It's just not the style that I want to do my current content in. Yeah, I'm not gonna spoil what the answer is, but uh, I will rule out smallpox and measles because I want to elevate sort of how scary I think this next virus is. The other thing I'll note is that I'm not like an epidemiologist, and this is a disclaimer that I'm going to make in the video too. I actually don't have enough background to tell you if this is actually going to be a threat to humanity. As a molecular biologist, all I can say is that there is a path where it is going to be very bad. Whether or not it is likely, that is more up to, your trustworthy health officials to determine, instead of some YouTuber. My whole thing is like molecules doing molecule things, and that does not give you an estimation of whether or not a virus is going to be a global threat to humanity. Yeah, so we'll just see. We'll just see. Yeah, we'll see. Um, yeah, it is something that I've been having a lot of anxiety over, and uh, it's something where like I've had a line of like I try not to make my videos anxiety provoking, uh, well because I don't I don't want people to click on my videos the same way that other biology YouTubers have their content clicked on because of like fear, and that might be hypocritical. You could say that like, oh, they clickbaity thumbnails and titles, although I don't think they're clickbaity or all that egregious. Because I immediately make it clear that I'm not going to be fear mongering in every one of my videos, but this next virus is one that I'm genuinely afraid of. What do I think of gain of function research? I'm not qualified to talk about risk and benefit of gain of function research. I'll just put it that way. That is, it is like, it's not necessarily a cop out because any good scientist worth their salt knows exactly where their expertise ends. And, I'm not even a qualified virologist, right? I'm not an expert in these fields. I just have like a very, I love reading about them, and I have a molecular biology background, so I'm comfortable in the language of molecules. I am not comfortable in the language of how diseases spread, what the chances of diseases spreading are, mutation chance, etc., etc. Thank you for joining the Helper T-cell, Leo. Thank you so much. Thank you, thank you. Very, very good. All membership money goes to a sitting fund that accumulates all of your membership money specifically to help me pay Toxo, who does all the art for the channel, because she will not take payment in any other form. And then I convinced her, what if the value of your work accumulated money in the form of membership money? Would you take a gift or a payment then? And then she agreed. So I will be taking her to a very nice omakase place once the funds have reached a certain point. And that is to say that like this is my part-time hobby. Don't feel compelled to contribute. Uh Although again every little bit does help, but only in a way where it just makes me happy. But thank you so much, Leo. If I ever do this full time, I will let you know, and then I will be a lot more shameless about having YouTube be a main source of income. But as for now, this is my hobby, and I am very happy to keep it that way. You could make something about immune amnesia with measles. I still think it's the coolest virus after rabies. That is interesting. I don't know a lot about measles, and the idea of immune amnesia is extremely tempting. As of right now, I think, go with my current plan just because for me it's a little more urgent to do this given current events, and so that I think is an interesting one to look forward to in the future. Would you cover treatments like gene therapy or only diseases and immune system topics? So I actually tried to interleaven my immune system pathogen stuff with non-immune system pathogen stuff. The one that I did before this was on the microbiome, which you could argue has a heavy immune system component to it, but like I also covered like tardigrades, for example, just because I don't want to be like the disease guy. I want to be the biology guy. So I will cover other things. It's just right now I'm doing 2 pathogens in a row. And depending on some grants that I'm waiting on, I might do a couple more pathogens in a row just because like it's much easier to get funding for science communication projects when you're doing something that has direct social good. And my pitch is that informing all of you of how viruses and bacteria work and advocating for things like vaccines and treatments, I argue that that is a social good. So you're seeing I'm feeding artists with this. That is true. And also thank you so much for the 5 gifted. That's amazing. Thank you so much. That's awesome. Feed Toxo, thank you. She will be very, very glad to have that goal reached very shortly. It's gonna be a cracked out omakase, by the way. Like it like in terms of, because I want to be very clear with my audience, right? Like I don't want to make it seem like I need the money, because I think that you should be able to spend your hard-earned cash in the best possible way for your own enjoyment. So I am essentially banking and slowly accumulating all of the membership stuff for like the dankest omakase that we can afford. Like, so it is a very long-term goal, like a very long-term goal, but every bit does help. And again, making it very clear, it's purely for our enjoyment. We do not depend on the income at all. But again, thank you so much. That is very exciting. That's awesome. Could you include some structural biology stuff in the future? I think it's neat. So I covered some structural biology stuff with Clockwork in a video that I did with him. That was a protein, Molecules of Life Protein 101 stream, which was very fun. If you want structural biology stuff, I have to say that I do not do a better job than Clockwork. So go and Google Clockwork Show. Yeah, like he's a really talented animator. He's also very funny and has great on like screen personality, and is much more capable of making structural biology more interesting than I will ever be. So there's my plug for Clockwork. Me, I'm more of like biology narrative guy, right? Pathogens are characters, the immune system are characters. There's a story behind all of it and how they operate. Whereas Clockwork, if you want to get deep into how molecules actually function, Clockwork is your biology content creator. A personal, heavy endorsement from me. So I think that would be wise for you to enjoy your content. Yeah, his stuff is actually amazing because he puts so much delicate care into how like animating everything. It reminds me of like the animations I used to watch to study, but like way better and a lot cuter. And he's also just a fantastic guy in general, like a fantastic friend of the show. Like we pass some resources to one another. I've had him contribute to a couple of projects of mine. And yeah, he's just awesome guy. Subscribe to him. Will you ever cover something kind of more banal, such as the common cold and how it stays an inconvenience and is not dangerous? I think that, so this is something that I've come to realize, and I've repeated this a couple times on stream, but I think I finally have the perfect way to say this. At first, I thought that I was going to run out of content because there was only so much that I knew about how viruses function. But the more I study about viruses, the more that I realize that every thing that you Google into Google Scholar, every virus name you Google, I'm 99.9% confident that there is going to be an immune evasion mechanism that is worth making a 5 neutrophil video on. Why do I say this? At first, I was reluctant to do Ebola because when I first read about Ebola, it was just like, oh, it's just interferon inhibition. That's not interesting. And then I revisited Ebola and found out a whole bunch of other crazy stuff. And I've come to realize that here's the thing. Your immune system is great, right? Your immune system is awesome at what it does. It has kept you alive for how many years? 300 million years as your Homo sapiens self, and a lot longer before you were Homo sapiens. A virus has to evolve some crazy stuff to be able to survive your immune system. It has to. There's no way that a virus survives by not doing something absolutely drastic and bonkers. So something more banal like the common cold, absolutely probably worth covering. I'm confident it's doing something crazy. I hope the grant works out. There's another channel called Science IRL that covers botany with science communication grants. Yeah, I'm really hoping I get some of these because it would be delicious, and I could also, invest some of that money into making the channel better, which I've been reluctant to do. All of the money that's come in from AdSense, I've been hoarding in a battle chest and kind of afraid to spend it, essentially. Because I like doing these videos on my own, right? Like, I don't have an idea of what I want the video to look like until I'm editing. So like hiring an editor, it can work, but like where do I find a combination that like an editor who wants to do editing part-time and is also a biologist, right? Like I could not work with someone without biology background because they'd have to already know what I'm talking about. Otherwise, the visuals won't come out correctly. And also my sense of humor, right? Like I don't tell jokes in my videos, but I do have a lot of visual gags that I like doing. Staying on molecular biology and fundamentals and viruses, could you cover endogenous retroviruses? Ooh. um, yeah, again, I'm not a virologist, so if you have suggestions, I'd be very happy to take them. Although, like, my content backlog is like so long that I don't know, but I'm not averse to shifting some priorities. So if you have a favorite endogenous retrovirus, that would be cool. What about anilovirus? Never heard of them actually. Or autoimmune psychiatric things such as Toxoplasma gondii. I did do a Toxoplasma video for April Fools where Toxo herself came on to do some ASMR. Toxo would be good to do one day. I'm not ready to move into eukaryotes yet. There's like yeah, when I do the ones I find, because like right now, because of the science communication grants, I have like heightened sense of urgency with regards to picking stuff that has larger implications for public health. Toxoplasma, yes, implications for public health. These next couple of videos I have planned are like potentially population level, potentially country level kind of little buggers. So 300 million years of Homo sapiens is a good one. We're in the Carboniferous and still at the point. Yeah, yeah. Laughs and vesicular stomatitis virus that just goes brr. Oh, well, I'm excited to learn about that then. Very exciting. It was a total joke video though. Like it's not very in-depth. I eventually will probably do a toxoplasmosis video, just not before I take care of the scary like humanity the humanity stuff, right? And I'm not gonna say humanity ending because I don't think any of these are humanity ending, but in terms of like, you know, avoiding tragedy is good. Is it polio? I'm not gonna say. I will eventually reveal the virus once I have the script fully ready. Yeah, because there's still time for me to decide that I don't want to do this because it runs so counter to my regular thing. Because like, if I wanted to do trendy stuff, then I could, but I actually purposefully kind of avoid doing trendy stuff because I want the joy to come from learning and not from taking advantage of people's current anxieties. But because of public health, some of current anxiety might be warranted. Anilovirus is really cool. It seemed to be benign, but nearly all people have it. Those are the most devious viruses, right? Viruses that are so good at evading your immune system that you don't react at all are I think, quite interesting. One of my favorites that I have yet to cover is cytomegalovirus, human cytomegalovirus, and the herpes viruses are in this category as well. So not viruses that you would typically think are like population ending, but viruses that are incredibly clever about how they go about evading your immune system. For humans, the W family fundamental for Synictin 1. Oh, is that the retrovirus that gave us our placental protein? Older elements basically work at TF binding sites for a huge chunk of our innate immunity. Interesting, interesting. Yeah, every virus has its own story to tell. I think they're very interesting. I can't wait to cover natural fungi too. It's just that like in terms of, well, you have to like triage, right? Because fungal infections are responsible for a lot of world misery, and so fungal infections will be on kind of like the medium high priority. But because they are slow to spread, I'm okay with delaying covering a fungi. Whereas viruses, it's like, oh, you know, reading the news, I'm like, okay, viruses I should cover now because viruses are very quick to spread. Yeah. Genuinely would love to hear about the human virome. I would too. It would be tricky just because, you know, I'm a meat and potatoes molecular biologist, and omics stuff, ohm stuff is stuff that I don't have that much expertise in, and so it can be quite tricky. Although Resta, if you haven't checked out my Bioinformatics 101 with Resta Makarabe, Resta is a bioinformaticist. And so covering a little bit about the basics of bioinformatics with her was a lot of fun. I think we covered a computational approach to tackling the cholera bacteria. So not the virome, but similar techniques used. And then with myelin, I covered the microbiome with some omics level tools. But virome, have yet to cover that. Yeah. Like all the viruses that are part of your microbiome. Oh, I see. Yeah, like the phage and stuff like that. It'd be cool to do a phage omics stream. But again, like in order to make this kind of stuff compelling, I have to be good enough to begin with. And for something like omics, it's tricky. Like with myelin, compared to me at the very least, is an expert in statistics. Me, I'm a real t-test kind of guy where I don't do a whole lot of complex statistics myself, but for omics and stuff like that, I would need a partner who is good at statistics, math, and programming to make it feel like I am making content to the standard that I want to make content to, right? Like if I don't find my own material compelling, like if I can't wrangle a good story out of the paper, it can be kind of tricky, right? Because the other thing about like just understanding what phages are in the body, so I do a stream on that, and then what's the story? Yeah, it's hard to imagine what the actual story behind that data would be. Not to say there wouldn't be one, but I have trouble imagining. Whereas something like viral infection, there's an obvious story. Virus does this, immune system does this, virus does this, immune system does this. There's a very natural storyline. But for omics data, it's like, okay, so we have this virus and it's present in this person. Okay. I would have to know a little more about how to make that kind of data meaningful. I know it's a bit more trendy, but would you ever cover what we know of the fish spitting near Florida? I have no idea what that means. Would you ever cover bigger stuff like malaria? I personally know a malaria researcher. So I have yet to coordinate with them. I would love to have them on. They're great. And that would be very fun to have them on. I would actually genuinely love that. And malaria is also crazy in terms of it being unlike anything that I would have covered up until this point. For those of you who don't know, malaria is a disease that hopefully you know is spread by mosquitoes, but malaria has an intermediate host, and that it is a mosquito. So it's not only adapted to infect humans, but it's adapted to infect mosquitoes. And it does things in different organisms. And, you know, there's a whole bunch of interesting differential genetic programs that it turns on depending on which host it's in. It's also more gory than bacteria or viruses because you get imagery like, you know, little protozoa replicating in your liver. For some reason, that freaks me out more than a virus replicating. Eyes emoji, Mylan, is that you volunteering for Virome stream to lend your statistical expertise? I appreciate you wanting to be a biology channel instead of a disease channel. I like your videos and the topics are always interesting. Keep on doing what you're doing. Thank you very much, Jaria. That means a lot, especially because I don't have to do this for a living. That I can enable this choice. And eventually, like, again, if this becomes my full-time job, it will be on the backs of membership instead of the AdSense revenue, right? Like, you know, I'm the type of person to plan like a decade ahead in terms of like where I want my life to be. And in a future where I don't know, get fired from my job or let go or something, or, you know, or if I have to move, in a future where this situation is true, if I do have to come to rely on YouTube money, I would like to have an audience that understands that I might not always cover what the algorithm wants me to cover, but rather what I, the human cell, human cell, the neutrophil that I am, what I would like to cover, because I hope to establish that relationship with you all where you trust my taste in interesting topics. So thank you. Those words are very, very nice. And of course, the membership support if that grows enough to the point where I can do this full time, that will be what I go off of instead of YouTube analytics, right? Because AdSense, deciding to go full time on AdSense is a recipe for pandering to the algorithm, which I do not want to do. So your messaging to me by doing membership, that, you know, in like a half decade future, you know, I'm not promising anything now, but as membership expands, if it does, that'll let me know if I even have the option to do this full time. It is a species of Candida that has been causing those deadly fungal infections recently. There's something that I find morbidly fascinating. This apoptosis, not apoptosis, this phagocytosis video that I keep on showing on my channel, that is footage of Candida being eaten by macrophages and then exploding out of the macrophages, making hyphae and stuff. Candida and fungi, they're, you know, again, like malaria, very, very creepy. In the sense that when I visualize them, I'm more reminded of a horror film or you know, like a horror video game than I am with viruses and bacteria, weirdly enough. I know there's only a few and little data, but what about virophages? Virophages, so these are viruses that infect other viruses, I believe, right, Joel? So for those of you who don't know, there are even smaller packets of genetic material that piggyback off of bigger viruses, and they actually need to co-infect with those viruses in order to have productive infection. We also already use one of these helper viruses called AAV, one of our favorite genetic engineering tools. I don't know if AAV counts as a virophage, but it is certainly a parasitic virus. Cryptosporidium, don't know what that is. I should have a good chance to plug the Discord. I should have a Discord channel where I solicit suggestions that are probably lower in priority than my member suggestions, because I've not been great at fulfilling those requests. Let me go ahead and plug the... I should set up stream elements to do that. It's probably a good idea. Go ahead, and if you haven't joined the Discord, here is the link. Oh, why is it not doing that? Give me a second. That's interesting. Is it because I'm in streamer mode that I can't invite people to the Discord? That's weird. And my moderator isn't here to do that. That's so weird. I think once I installed stream elements, maybe things got a little funky with regards to Discord invites. Maybe I'll post it somewhere. Yeah, unfortunate. That's not working. They don't seem like they could be engineered to solve our viruses, like the flu or COVID, as they only have been known to infect larger viruses. There's an idea that I wanted to do for months now, which is to do like the gargantuan gigaviruses. Oh, Toxo, thank you so much. Oh, what good timing. What good timing. Thank you so much. I don't know, like my Discord won't allow me to paste the link, which is really bizarre. Everybody say hello to Toxo. Toxo, we got 5 gifted members today from Leo, who also became a member themselves, which is very exciting. So that's contributing to the Omakase Fund. Cryptosporidium is a pathogenic amoeba, usually in poorly maintained pools, and I hope you never have a brush with it. Leo is feeding me. Yeah, it's true. Very cool, very cool. Very nice. Yeah, I'd probably cover this Cryptosporidium. Is that contain the brain-eating amoeba too, because that will probably be the big video of 2026, right? Like Yersinia pestis, probably 2025, brain-eating amoeba, probably 2026, and then whatever viral sensation that people endlessly want to know about, 2028. I don't want to make it seem like I'm like begrudgingly covering them. I genuinely want to cover them, but I'm afraid that, you know, any YouTube channel that covers a whole bunch of trendy things all at once and then you stop covering those trendy things, people will stop watching. And I would rather have my strategy, my YouTube strategy is more to grow this segment of my channel, using almost the videos, which are my main passion, but I can't do my main passion sustainably if I ever have the dream of doing this full time. I cannot rely on the popularity of those videos. And so for those of you waiting for Yersinia pestis or brain-eating amoeba, please understand that in order to grow organically, sustainably in the long term, that I am purposefully waiting to do those. Which is kind of like a risk, right? Because there's a chance that some other channel that does something similar like mine props up, but this is a risk that I think I am happy taking, right? If someone else wants to splash on the scene and cover biology in the way that I cover it, I wouldn't be mad. But the motivating factor for me to keep doing what I'm doing is that as big of a head as I have, I think I am the best in my own niche. In my very specific niche of animations regarding higher level order molecular biology processes behind pathogens, this very tiny niche, I think you have to be crazy to want to do that to make YouTube money. So I think for now I'm pretty safe, because otherwise, like, you know, there are other channels that are going to explain these things to you, but there's just like guys talking to microphones, which is fine if you don't want to wait for me to make a video, which is also fine. But I kind of want to bank on my self-confidence of me having a unique lens and a unique storytelling strategy that I hope you all enjoy. Let's see. What about Kirkazad? The thing is that I used to view Kirkazad as competition, and I was really bummed when they released their rabies video, because it was right around when I was going to release my rabies video. You can check the timestamps. I actually had to, I think what happened was I got so bummed out that I never published it and then redid it for Fight the Neutrophil. But I've come to realize that when I watch Kirkazad videos, the animations are spectacular, but I don't think their goal is to be as thorough as me. Also, I think Kirkazad, I think what I want to rely more upon is I don't feel like I come across as a polished science communicator. I think I am measured in my speech, but I think that's different than being a polished science communicator, right? Because I would prefer you see me as a guy that has research background, that is really passionate about what he studies, and there's more of a personal connection of like, I'm geeking out and I'm geeking out with you, as opposed to Kirkazad, where it's a company of researchers and contractors and animators and editors and board meetings and script meetings, and the sense of like, this is a product that was carefully crafted for you. That's not to say that I don't carefully craft my videos, but for me, I think part of the Five and Neutrophil experience is that it is my love that I put into this. And I think that that love cannot be subsumed by a bigger channel with a bigger budget and a tighter presentation style. Hey, am I late? We are at the very end, unfortunately, but we are just hanging out and chatting right now. Kirkazad started yapping about politics. Kirkazad, you know, that's something they still do very good health videos. Like, I think the vaping video they did was quite good. It's also why I've never touched marijuana. Fun fact about me is I'm like aggressively straight edge. Didn't have like the thing is, I'm straight edge, but I'm like lawful good straight edge, where it's like even rules that don't make any sense, I still follow because I am a cog in the machine, you could call it. I'm not a rule breaker in any kind of way in my personal life. Like, I didn't even have, like, all of my alcoholic drinks were legal. I might not have had an alcoholic drink. I've had alcoholic drinks before the legal age, but they were approved under chaperone laws. And so every drink that I've ever had was fine. Which is kind of an arbitrary rule to enforce, right? With that being said, like, part of my fear of marijuana, even though it is legal now, is that there was research showing correlation between marijuana usage and psychosis. And you know, the vaping situation kind of echoes this kind of like unknown risk that people take when they do consume these products. That is to say, alcohol also very bad for you. You know, that is a known level of badness. But for some reason, just like my personal assessment is that things that affect the brain in that way were more terrifying to me than something like liver cancer, which it shouldn't be. But, you know, viewers of my channel know that I personally value afflictions of the mind to be spiritually more important for me to hold in high regard, right? That's why the first cancer that I will cover in detail is a cancer of the brain. That to me, the mind is so essential to your personhood that anything screwing with the mind long term, in something like psychosis or schizophrenia, that to me was no go, off limits. So even though where I live it's legal, I will not touch that. And I've also reduced the amount of alcohol I've drank or drink to like a very, very, very tiny amount. An occasional sip of alcohol when I play Dungeons and Dragons is about all I will allow myself to have. Goody two shoes bros on top. That is me. That is me. I am very much a rule follower. Even if I feel that the rules are arbitrary, I am a square peg to be fit in a square hole. I actually joked about this, not only in V-Theorem, like in our private Discord, but also just like to my coworkers, is that I will like lick any boot if it means that I get to keep my job. So like, just know that about me. I am not a counter-revolutionary, despite me being like, oh, you know, conventional science communication. I want to go against that. I do so in my safe box. Fi is a very lawful good. Yeah. Toxo knows. How did this turn into underage drinking? I'm just saying how I'm a rule follower. I'm a rule enjoyer. On the topic of alcohol, why do yeast cells decarboxylate pyruvate first and then reduce it instead of reducing it immediately like other cells? That's a question that you might have to ask a yeast researcher. I don't know that answer myself. I just realized how many ways killer T cells can die, more than 10. Absolutely. There are so many ways that cells can go out. Although in your canonical biology education, you really only hear about apoptosis and, if you're lucky, necrosis. There's all sorts of other autosises that can happen, like ferroptosis, which is death by iron signaling. Stuff that I never actually even knew about until I started doing research for Fi the Neutrophil channel. Hey, Fi, loved your immune system video. Thank you. I worked really hard on that, and the algorithm did not appear to agree. I still think it's great, so I'm not too upset about it. But it was another video. Like there's so much love that I put into every Fi the Neutrophil video that it's hard for me to pick a favorite or one that I loved working on the most. Although I do have a least favorite. My least favorite video is probably the kombucha one. It flopped so hard, and not only did it flop really hard, which contributed to my distaste for the video, but also I just don't think it's a very good video to begin with. That was my first, like, I would like to ride the algorithm, so I will put Brad Leone's face in the thumbnail, and that turned out to be a quick revelation that I should just stick to what I genuinely want to cover instead of chasing trends. Thank you for renewing membership, Trojanet. The various tosises would be an interesting stream. That would be a really good idea for a stream, like a tier list. a tier list of tosises. That would be really fun, I think, to rank all of them. It would also give me an excuse to learn more about them because, you know, again, apart from apoptosis and necrosis, I actually don't know a lot about how the other cell death programs work. That'd be cool to learn together. I really enjoyed the stream. Thank you for making this space for discussion. No problem. I often find that, like, you know, in terms of my position in grad school, I was like in the middle of the pack in terms of like grades as a proxy for like how smart, quote unquote, I was. But I've always felt that like I subjectively felt that I was on the lower end of complex with my line of thought. And I think that made it so that I was maybe a little unsuitable to be a full-time scientist. Complexity of thought meaning like I prefer to think in ways that I could communicate to a non-expert. It is my domain that I prefer to exist in, as opposed to talking to other scientists about my science. And I think that's been an asset because I love talking to you all. So thank you for thanking me for making the stream, the space for the stream, but also thank you for showing up and engaging so thoroughly with the discussion. Could you make a video about variola? So variola, remind me, that is chickenpox. So that has been on the list since the very beginning of the channel. Right, variola is chickenpox. Yeah, from the very start, chickenpox was going to be something that I covered because I myself got shingles. It was my most stressful semester, and I got freaking shingles, dude. And so that particular method of immune evasion, where you are exposed early on in life and it hides in your neurons until they're ready to become shingles, that's also very interesting. Um, or is chickenpox varicella? It's variola, right? Let me Google that real quick so I don't spread misinformation. Variola viruses. Which ones are these? Oh, smallpox. Ah, okay. Which one is chickenpox? Smallpox will definitely be covered. Varicella. Ah, you can see why I got confused. They're both very similar sounding. Got chickenpox as a teen. I need to convince her to get shingles vaccine. Please do. Shingles. Luckily, Toxo saw it on me quite early. Toxo knows what a shingles rash looks like. Thank God. But it can be very painful. Very, very, very painful. And debilitating. You know, like you can't move painful. And Toxo caught it on me just in the nick of time, and all I needed was a simple antiviral course, and I was fine. But I'm too young, despite referring to myself as a hag. I am too young to get the shingles vaccine. So if your mother is not too young to get a shingles vaccine, it's worth getting if you are eligible for it. I also have shingles. Yeah, it's interesting. Shingles has actually been on the rise in younger populations. Shingles was known to be a disease that old people get when their immune systems are compromised and the chickenpox virus reemerges. We're actually seeing a global trend where the onset of shingles is decreasing. So when I got shingles, we were like freaking out. We were like, how could I be that old? I am not that old yet. But globally, there's a trend and no one knows why. So it's pretty interesting. Yeah. I had chickenpox as a kid back in the day. Get yourself or your kids vaccinated. Yeah, yeah. I was not vaccinated. It was unfortunate. Back when I was a kid, it was not as easy to get or commonplace than it is today. So if you have the option, it is worth getting for sure. Changing the subject, but how did you get your art style? This is Toxo's art. I don't know, Toxo, if you're still in chat, but maybe one day it would be interesting to do a behind the scenes as to how you came up with such good cells. To say the choice of design, we back in COVID days wanted to like make Cells at Work before Cells at Work came out, but then Cells at Work came out and it was way better than what we could do because of course Cells at Work is made by an animation studio and a whole bunch of professionals. Just two people in a small apartment room were not going to match that level of ability. And so Fi the Neutrophil is a character. Well, the Neutrophil is a character from early sketches of that animation that we were making. And so instead of doing an animation, because we could never be as good as Cells at Work, what we can do better is being VTubers. So Fi was then taken from a 2D animation into the VTubing space by Toxo when that was something that I wanted to do. Both on you and Kirkazad made me a med student. I'm screaming in joy. That's awesome. I love to hear that. That's so great. Although I never pursued medicine myself, but it is good to have more young people in fields where we are fighting for the collective good of humanity, right? Like to sort of break from talking specifically about biology, the enterprise of science is a human focused endeavor. And so anybody that we can get that is working to combat, pathogens, whether it be through research or medicine, it's worth it. It is good for humanity that we have people who are interested in this kind of thing. The droopy dendritic cells got justice. Yeah, that came out of I think I said, oh, it would be fun to have like an eldritch kind of design for dendritic cells because they are of course dendritic, they are branched, and then Toxo came up with a very droopy boy, and I love all of Toxo's designs. So I guess I could say what the process is that I will give Toxo a very short description and then Toxo's brain will just come up with cool stuff. like the macrophage also has a bit of eldritch vibe, because if you look at what these look like under EM, they're all kind of similar. They're all kind of goopy, you know, whatever. Um, but then Toxo like cutifies them to make them more character-like, and I really think that those are answers that I cannot provide. It's just Toxo's brilliance. Toxo's decision to make Ebola a two-headed dragon was also very fun. Yeah, Ebola, of course, does look like a filamentous pretzel-shaped virus, but, you know, as she was drawing it, she was, yeah, it kind of looks like a two-headed dragon, and then that's what it became. It's very cool. I've been feeling out of my research mindset due to stressors, but this stream is really motivating me to go read some papers today. That's awesome. I recommend just going to nature.com and just picking a paper. Cell is also fantastic. Those are my two favorite journals to read just for this channel. Plus, I would also recommend if you want to challenge yourself. But Nature and Cell, those are my go-tos for easy casual reading. Do you consider to make the B cell match with all the other cells, like the wobbly thing at the center? Oh, the nucleus? This is something that Toxo came up with, because all the other guys have nuclei in the center, as Estella says. But Toxo was like, B cell doesn't feel like a fella, because we refer to all the cells as fellas. And she was just like, the B cell doesn't feel like a fella. I was like, what do you mean the B cell doesn't feel like a fella? She was just like, I don't know, the B cell feels like a thing. It doesn't feel like a fella. And I was like, why? So the mysterious process by which Toxo designs these, I cannot explain why the B cell was singled out as being not a fella, but the rest of them are. I guess that's just a little bit of Toxo lore, is that her creative mind works in very mysterious ways. Oh no, the B cell is a place, not a fella. Oh, gotcha. That makes less sense than what I said. That flat out makes even less sense than the thing that I said. But yeah, apparently the B cell to Toxo was a place and not a fella, and that's why it doesn't have a nucleus. This is where the magic is, though. Yes, I don't think this channel without Toxo would have just been me in a lab coat talking to a camera. So Toxo has really helped to elevate the presentation of this channel. You know, I had my own podcast before I ran Find the Neutrophil, and it was pretty cookie cutter. The main appeal of me, I think, is me, but sometimes me is not very appealing. And I think Toxo has been invaluable in terms of elevating this channel to where it is today. What is a cell if not a place that moves around? But what about the other cells? Why are the other cells fellas? In fact, like, B cells arguably don't even move around that much. Neutrophils and macrophages are much more mobile. What is my opinion on Sci-Hub? Almost everyone I heard from is for it, except people for making money from the paywalls. I'll say this very shortly. Academic publishing, as much as I love Nature and Cell in terms of the quality of paper, I do not approve of their publishing practices. You cannot imagine how difficult it is for me to share what I have learned, because as I showed in this PowerPoint, like one of the first slides, I had a paywall right at the start. Right here. The preliminary data, not preliminary, but the data that shows that Ebola doesn't infect T cells, data copyrighted, can't show you, even though the research that your tax dollar pays for is funding this information. So why the hell doesn't the taxpayer get access to this data? It's impossible to understand. So I'll say that it sucks that Cell and Nature are so good to read, because their publishing practice, I think, not great. So that does mean, plus, if you want to, you know, advertise, I'm already doing it for you for free, but if you want an official kind of plug in a full-length Find the Neutrophil video, anybody from PLOS, if you want to reach out to me and have me be the poster boy for open access research, please reach out. I would be very happy to do you a sponsorship. No, I can't explain why, but Toxo's logic around B cells makes sense. What are you all saying? A fella can be a place for bacteria, so let's just scale that down. I mean, I guess it's about the vibe. What vibe does B cell have? B cell is a war criminal that sends out unmanned drones. That's very funny, because that is actually a part of the script that I have for my next video, Akazahedron. That is literally the comparison that I am about to make. That is very fun. Maybe it's because the B cell's entire goal in life is the factory must grow. I don't think Toxo's thought process went that deep. I think Toxo, if I am lawful good, Toxo is chaotic. Chaotic good? Chaotic? Definitely chaotic. Chaotic good? Chaotic good. I think chaotic good, but chaotic. We pay to publish in PLOS so you can read for free. At least there is a mechanism by which the taxpayer can read their own taxpayer dollar funded research for free. So while that is still kind of a sucky way to do things, it is, I think, fine. Especially if the university has funds to pay for that kind of thing. Publishing itself is just a mess as a whole. And I don't know what can be done about it. But I can continue to advocate for open access policies like PLOS. I'm changing the subject to art again, but when will we have more lore? Or I guess how do I explain rather than art literature? More 5 the neutral lore stuff. I don't know if we can say right now my name, but let's just say that there are extra 5 the neutrophil stuff that is on paper a thing that might not. I don't want to promise too much and not deliver, right? Because there are plans that are very loose right now for more 5 the neutrophil content that doesn't have anything to do with viruses or pathogens or science, but just fun stuff that is just for Phi. So it is in the works, but whether and when that is released, who knows? Whenever my wound hurts, I just think these neutrophils are burning everything in sight. SMH. They're also helping you repair, so, you know, it's important that that both happens. It's fine, just 1.9k out of my own pocket. Whoa. Okay. Is it your own, like, lab pocket or your own personal pocket? Like, that, I think, is a difference, because I never made it far enough in academia to publish myself, so I'll say that my perception of these journals is purely from a secondhand perspective, like hearing other people's story. Personal pocket, don't publish as a lecturer. I see. Yeah, that sucks. And I assume your institution doesn't have like a slush fund for that kind of thing, and that blows, and I'm sorry. But at least PLOS is cool. At least PLOS is a good journal to publish in. Anybody would be proud to publish in PLOS. So at least you are paying, I guess, for that. It'd be fun. 1.9k to publish. Yeah, I mean, I don't want to like cover this in an official mechanism just because I'm not like a very I don't have a lot of first-hand experience with the publishing system, so I can't say all that much. What I will say is that like every time like Nature or Science has an editor come to our university, my old university when I was in graduate school, to like give a talk, they would get grilled by the PIs. Everybody would be so mad, and PIs get mad in ways that only academics can get mad. Like if you've never heard a passive-aggressive PI before, it is a marvel. Like I think that my language is quite academic, right? Like picked up a lot of my speech habits from the very highly educated people that I was surrounded with in graduate school. But when a PI wants to be condescending, rude, angry, confrontational, they generally have it in a way where it is so specifically passive aggressive with big brain language that you can't even be mad at it. Like it's something that commonly gets tossed around in American culture is bless your heart, right, from the Midwest. But bless your heart sounds condescending. When a PI is condescending, you might not even realize it's obfuscated through so many language barriers. And then you think about it, you're like, oh damn, the PI went hard on that that journal editor. It's crazy, and a lot of fun, and you know, very snide, which is great. Passive aggression in academia is a whole thing. It's wild. We should have a stream where we just talk about stories about passive aggression in academia, because it is fun, but it's fun in a way where like bless your heart, to me that just triggers something annoying in my brain where it is immediately anger producing, which isn't fun. But like academic passive aggression can be entertaining because it is so wrapped up in the language of academics, which I think is a good time. Yeah, I definitely saw angry PI in my time doing undergrad engineering research. They just disassemble whatever made them mad. Engineers are a different breed of folk too. Like I think there's academic weird, and I think engineering academics are a different kind of weird. They are um uh they've got their own subculture within academia. I think that's fair to say, which I think is very very fun to think about. All right. So my voice is getting a bit parched, which is actually sort of worrying because how long have I been yapping for? 3 hours. So this glioblastoma stream starting on January 11th at 9 a.m. EST, I will be going for 4 times that length in a 12-hour charity stream. And I'll yap a bit so that you can use Stream Elements chatbot to get a little information, more information about that. But essentially, I'll be covering glioblastoma, this aggressive brain cancer research from 9 a.m. to 9 p.m. And I'll take breaks, and during that break, those breaks, I will be completing some milestone rewards. So like things like making you play ink shorts, stuff like that. Those incentives will be gated behind certain donation incentives, and also if you donate singularly a large enough value, so $10 USD, if you donate at least $10 USD, you can make me spin the good wheel, which isn't actually very good. And then I will do those challenges during my break times. So things in the good wheel can range from push-ups because they're good for me, squats because they're good for me, drinking a smoothie made of the top 5 superfoods, no matter what those superfoods are. Technically, that's good for me. That's why the good wheel isn't all that much fun or good. And then for $20 USD in a single donation, you can make me spin the bad wheel, which has all manner of horrible challenges, including very spicy things, and then like having to talk about science with those spicy things. So those are, I think, definitely going to be interesting and will help keep the very long stream fun. If you are interested in cancer biology, one of the donation incentives is to make a true Find the Neutrophil video for a cancer. I've been reluctant to cover cancer in the past because there is such a heavy emotional burden with regards to covering something as serious as cancer. Cancer is common enough where everybody will know someone who has had cancer, and then chances are not bad that you also know someone who has passed from cancer. And so doing like little visual gags has never sat well with me, but I think I am practicing the art of balancing comic relief in the true sense of relief from the dread that is cancer and treating cancer with the appropriate amount of levity which it demands, essentially. I'll be covering in the first 3 hours a background on cancer as a whole. So if you are interested in Cancer 101, make sure to show up for that time slot. Of course, you can rewatch the VOD. I'll maybe consider chunking it on my second channel, Find the Neutrophil, maybe, which you can find in my channel description, not the video description, but if you go to my direct channel and then scroll all the way down, you can find the other members of Ethereum channels, links to their channels as well as my second channel. But a donation incentive that I think would be really good, one that I would be very happy to do, is to finally make a cancer video for Find the Neutrophil, just because, just to sort of tease what's coming on the 11th. Cancer is the worst. In terms of a filter to create the most dastardly thing that could exist, all viruses, pathogens, all of those pale with the gravity, the intensity, the severity of the mechanisms behind how cancer propagates. And to tease that a little bit, to indulge myself in talking about this just a little bit, it's been said, and you've watched Cells at Work, hopefully, but Cells at Work is, makes this argument that cancer takes a very sympathetic viewpoint to cancer, that cancer is a cell that does not want to be eliminated, and that is true. And so when you watch Cells at Work, you end up kind of feeling bad for that cancer, because if you develop conditions within a cell that are ripe for development into cancer. There are 1,000 different ways the cell will kill that cell to prevent it from being cancer. Logically, you have to deduce that any cell that becomes cancer has evolved a way to outsmart all 1,000 of those defenses. Not even Ebola can do that. Not even the Spanish flu can do that. Not even rabies can do that, right? While rabies does have like a 99.9% lethality rate, we outsmarted rabies by improving our immune system. In order for cancer to survive, it has to be good enough to neutralize every single detail, every single plan that the immune system and the natural genetic code of the body has in store. And that is why cancer is so devious, so devastating, so severe, so complicated, is that the number of things that cancers can do, nothing compares. And so to make a full video about that would be a wonderful incentive donation if we can meet it. As you are a neutrophil yourself, what do you think about the significance of neutrophils in viral infections? I don't know enough about neutrophil involvement in viral infection, despite being a neutrophil myself. Perhaps I've never encountered viral infections on my own in the wild. So far, I'm not very impressed. So far, it seems that neutrophils don't seem to do all that much, although I could be proven wrong, depending on what viruses I choose to cover in the future. I would love an academia story stream, and I'm an undergrad with no experience. Yeah, that would be fun. That might happen sooner rather than later. It'd be fun to talk to my fellow V-Theorem academics to see what their experience in academia was like. A billion fail and one goes by undetected. Exactly. Like that, I don't think was hit hard enough in Cells at Work, is that the immune system is really, really good at killing cancer. It's really good at killing cancer. It's evolved a specialized cell called the NK cell, whose job it is to kill cancers and viruses too, but specifically it can kill cancers. And the thing about cancer is that most of them die. And so in order to truly become cancer, you have to be crazy. The cell has to be doing some absolutely insane, crazy nonsense Deus Ex Machina level evasion of the immune system. And that is what we will be going over on 111. And if we can meet the donation incentive in a real Find the Neutrophil video. I have a tense admiration for cancer as someone who has had to work, with it and think about and diagnose things that have been broken in interesting ways. Yeah, it is good to have people interested in cancer. It is good that people are motivated not just by the humanitarian aspect of cancer, but also the underlying mechanism of cancer, because you need passionate people to study this. You I think would be, it is okay to indulge in believing that cancer is cool because it motivates people to do something about it. It's okay to have multiple streams of motivation. I think neutrophils play a big role in sectioning off TB. From what I understand, that's a lot of macrophage involvement. I actually don't remember how big neutrophils were in the TB story, if I'm being honest. Maybe that could be true. I don't remember. I would love a video on how to get into research. I'm an undergrad currently at a community college with no opportunities. Community college is tough to get into research for sure. In some cases, it's just about emailing people and being okay with rejection, like the boomer thing of like, you'll walk into an office and you'll give them a firm handshake. But if you do that by email, and you are specific with your intent, then that is your best case scenario. Don't become one of these mass mailers who's just like, hello, I'm so-and-so student and I'm interested in your lab. Here are my details. If you are interested in doing research, your best case is to get in to an R1 research university where those opportunities exist, or an internship with a pharmaceutical company if what you want to do is life sciences. If you are at a community college and you don't have great opportunities there, it's a lot harder, I think. But if you want to be bold, and I mean, it's hard to get an opportunity at a community college during the academic year, but if you are off during the summer and you have the capacity to do research, you can try to look into opportunities that open up in R1 universities over the summer specifically. So if you are an undergraduate who can do an away rotation in a summer lab, that could be a thing. You're going to have to do a lot of research, though. It's not going to be easy. But when you do find those opportunities, read the researcher's faculty page and understand the research that they do and articulate why you think their research is interesting, and don't use ChatGPT. Come across as a real human being with actual interests, because a lot of academia will, for as big high profile as professors are, they're still reading emails. And so if you are going to reach out to research opportunities, suck up to them by saying how cool their research is. That's how I got all of my research opportunities. And it's not deceptive or manipulative. The person that you want to be hired by wants to know that you have thought about what they do for a living. So that is what I would say. Does the type of cancer give cells some kind of advantage? So we're going to talk a lot about cancer advantage from an evolutionary and mathematical point of view, game theory point of view. That's why I think that I'm very excited to frame cancer as game theory. It might tickle the bones of all of my programmers in chat, all of my math people in chat, because I know there are a lot of you out there who love programming and math and logic and stuff like that. And cancer is a case study in applying game theory. So look forward to that, Mr. Winter. That is something that I will talk at length in the glioblastoma stream. And again, if we meet those donation incentives, to make a whole video about. The current donation incentive, by the way, is $500, and we consider that a success. I derived this number from Rin Penrose's $5,000 donation goal, and Rin has 10 times the CCV as I do, so I divided that goal by 10. If we can get to 500, that'd be great. I'm also matching up to $110. It was going to be 100, and then somebody donated $10 on the stream that I was announcing the glioblastoma stream, so that I didn't want to make any money off of it. So I will be matching up to $110 for that stream, and I really hope to see you all there. If you don't have the money to donate, it's not a big deal, no worries there. Of course, you worked hard for the money that you do have, and so you have the opportunity to spend it in any which way is most valuable to you. But what I will say is that I really do hope that enough of you show up at least to hear me yap, because again, I have not covered cancer because it's not interesting, but because I feel bad about covering cancer. Now that I've found a way to cover cancer in the way that I want to cover it, it's going to be really, really interesting. At least I think so. Thank you for the advice. I've been looking out for summer opportunities, but also I've been thinking about just reaching out to labs at R1 colleges near me. I think reaching out to R1s is a good idea, but for a summer rotation, that's your best chance, just because unless your community college has partnerships with R1 universities, I don't foresee that approach really being very fruitful. You could also take part-time jobs at some research institutes if you want to just get exposed to research and you want to make a bit of cash. I know people who are working part-time in our mouse colony if you want to just get experience handling mice before you dive into research. But if you are like, I don't want to take a work detour, I want to do research, and I know I want to do research, then maybe don't do the mouse thing. But if you want to be like, I want to ease into the idea of working in a research environment, then applying for like a mouse colony manager, it's not a bad idea. Game theory and cancer, yes, exactly. This idea of winning and losing is important when it comes to understanding why cancer is the way that cancer is. Can plants get cancer? Absolutely. In fact, one of the bacteria that causes plant cancer, agrobacteria, is one of the most important bacteria in our agriculture business. So agrobacteria is a bacteria that gives plants cancer. And not only does it give it cancer, it also serves as a very convenient way for us to deliver certain packets of DNA to plants when we need to genetically modify them. So if you have a genetic modification in a plant, then you likely got it in there through the use of agrobacteria. I wonder how cancer correlates to autoimmunity. Excellent question. It's not something that I think I will cover in detail in the stream, but it is something that I will touch upon just because the cancer story is so messy. and complicated. And if anything, what I hope to get out of the stream, what I hope to impress upon you all, is this idea that the reason why we need so much funding for cancer research, why we haven't cracked this code yet, is because of its more near infinite complexity. So, something like the rabies virus, 5 genes, yes, deadly, but also not that hard to understand and compared to cancer, right? There are still mysteries to be solved in the rabies story, of course, obviously. But in terms of like, yeah, we got an antibody, we got a vaccine, we're good, dude. You get the vaccine, we're done with rabies. As long as you can get the vaccine, you're good. And on the research front, right? For actual casualties, that's more to do with government and policy and infrastructure. But in terms of science, rabies is pretty well taken care of. We've also conquered rabies to the point where some people use it as a genetic research tool to use rabies like you would AAV to deliver DNA when you need to engineer when you need to engineer mammalian cells. That's the topic of Science RL's latest video, agrobacteria and plant gene modification. Yeah. It's really cool, actually. Very cool. Cancer is basically infinite ways to skin 1,000 cats. Yeah, exactly. And the specific strategies that cancer evolves to take advantage of, there's stuff that I would not have even imagined in my wildest dreams. Like the Ebola story. If you watch the Ebola video, Ebola making VP35 to block STAT1 from binding important. That's classic molecular biology. If I were designing a virus, I could think of that. When it comes to glioblastoma, it's doing things that I'm not even clever. I'm not even creative enough to like think of how to do some of that crazy stuff. And yet cancer does, and it does because it has to, because if it didn't, it would have been dead. Things like viruses / pathogens that trigger cancer. I think what icosahedron is referring to is the fact that the immune system is actually responsible for a lot of cancer death. Your NK cells kill a lot of cancer cells, but with anything regarding the immune system, you also have the potential issue of autoimmunity. While there are viruses that can increase your chance of cancer, like what's the big one? HPV, right? I believe HPV increases the chance of reproductive tract cancers, and then there's that bacteria that I covered in the microbiome video that increases your risk for cancer as well. I think those are slightly different things: autoimmunity and the regular immune system. All right. Okay, cells and places. I need to go prepare for Christmas. See you in the new year. See you all in the new year. And with that, I think that is a great time to end the stream. Actually, Toxo just got home, and I believe Toxo has some boba tea to share, which is very fun. So my next stream will be this charity stream. I hope to see you all there. I'm very excited, and I hope you show up. Look forward to more Philo Neutral content in the future. And there's not a set date yet, but Myelin and Syzygy, who are two of the newest members of Ethereum. Myelin does social science, psychology, and specifically the psychology of fandom. So if you're into anime and statistics and psychology, go check them out. You can go to my channel for their links. I should make a Ethereum command where it just prints out all the plugs. That would be really helpful. And then Syzygy, or Z, as she is known, is an astrophysicist, mathematician, engineering VTuber, also making some very high quality content. And they are going to debut in January. So please look forward to that in the new year as well. And with that being said, thank you all for coming. I hope you all had a good time. And I will see you all in the new year. Bye. Bye-bye.